Okay, so we've got as you've seen, a very packed program. Our next speaker was not able to join us in person, so Giulio Vistoli, can you hear us? There's activity at the back. I think they're trying to connect. Very good. We can see you. So if...
Can you see? Can you hear me?
We can see you and we can hear you. Thank you.
Good.
So, please, you have...
I cannot share my screen until now, at least.
The screen sharing is... the slides are going to be shown from our end. So if you can just say, next slide, when you'd like the next slide, please.
Okay. You will use the...
Yeah.
Perfect. First of all, I'm sorry for my impossibility to be in Brussels today, and thank you for giving the opportunity to present my communication even from my office. The project is focused on the early case on SARS-CoV-2, as you can read from the title of my presentation. Next slide, thanks. The early case is a quite interesting target because along with other few proteins such as the RNA-dependent polymerase, it represents a minimal but fully functional replication of a transcription system. And so we can imagine that inhibitor of helicase can have a significant antiviral activity. Nevertheless, until now, very few inhibitors for helicase from SARS-CoV-2 were published in literature. And so this project tried to obtain a good number of helicase just to better understand the role of this protein. Next slide, thanks. The reason why until now there is a scarcity of inhibitors from helicase are several reasons that we will see in the next slide. And probably one reason is that this protein is quite complex because it includes two enzymatic activities, an unwinding activity, an expected unwinding activity, and ATPase or nucleotide hydrolysis activity. Of course, the two activities are connected, but the protein include more than one binding site. Indeed, from a structural point of view, as you can see in this slide, the protein includes 5 subunits, among which we can mention the zinc binding domain with 3 zinc ions, which are involved in the interaction with other protein in the transcription complex, and the 2 RECA-like domain. At the interface, there is, as you can see in the lateral figure, there is the binding site for nucleotide hydrolysis. Next slide. The project tried to significantly combine our expertise in performing enhanced and validated virtual screening campaign in the University of Milan, in my group of the Department of Pharmaceutical Sciences, with the facilities and the expertise in high-throughput screening from Fraunhofer in Hamburg. In detail, in Fraunhofer in Hamburg, specific assays to measure the unwinding activity and the ATPase activity of helicase were developed, and these assays can be implemented in a high-throughput screening condition. And so the synergistic combination of these two activities can provide some important outputs, which are listed in this slide. Of course, we can imagine to obtain a good number of new and improved hits, and so hit identification, including probably new chemotypes and new scaffold. Of course, the availability of a good number of inhibitors and new data can allow the generation of improved predictive models for virtual screening, and more in general activity profiling. We can explore the role of allosteric cavities, because I will see in the next slide the protein also include allosteric cavities. And in general, we can better understand the enzymatic activity and more in detail the inhibition mechanism for this enzyme. Next slide, please. Until now, there are a limited set of resolved structure for this enzyme, and most of them come from a study in which a good set of fragments were co-crystallized with the enzyme. And so, by analyzing the distribution and the location of these fragments, we can individuate, we can clarify the position of the possible binding site. And so we can recognize the ATP binding pocket and the interface between the two recalite subunits, as I remembered in a previous slide, the RNA binding pocket, and at least one allosteric pocket, which is between the zinc binding domain and the remaining part of the protein, the stalk domain, which is the so-called stalk pockets. And in our virtual screening campaign, we used a specific crystal structure for each of these three binding pockets, and we analyzed all these three binding pockets in our docking simulation. Next slide, please. At the beginning, we performed some preliminary virtual screening campaign in order to validate our computational strategies and to better understand the main feature of this target of this enzyme. And to do this, we collected the typical database in which we included the 1% of active compound mainly taken from literature that were combined with the 99% of inactive compounds. Few compounds, inactive compounds, of course, taken from literature, and most compounds are decoys collected from typical database of decoys, such as DUD or Decoy Finder. Next slide, please. And in just a few words about two approaches that we implemented in our virtual screening campaign, since we implemented the so-called binding space concept, according to which we considered in our virtual screening analysis not only the best pose and so the best score as usually done in a virtual screening and docking simulation, but we consider all the poses, usually 10 per ligand, all the poses produced during a docking simulation, and the scores obtained can be handled with several statistical parameters, and in this virtual screening campaign, we focused attention on the score means, namely the average of the score values for all the poses that we generated for each ligand. Next slide, please. And the second approach that we implemented is an approach to obtain a consensus function to optimize the performance of our virtual screening campaign by combining different scoring functions and different molecular descriptors by using this algorithm that we developed, the enrichment factor optimization, by which we are able to generate a linear combination of scoring function, and these linear combinations are generated by a search algorithm, which is represented in this flow chart in the left part of my slide, a search algorithm which is driven by a quality function which is substantially based on the resulting enrichment factors. And you can see in this study in which we validated the role and the potentialities of the enrichment factor optimization, that this method can provide very good result in typical benchmarking analysis. Next slide, please. And so, concerning virtual screening, just a few words about the methods. We utilized the plants as docking program by generating 10 poses per ligand, which were scored by ChemPLP. Then for each pose and each result, we generated a set of scoring functions by rescoring analysis by using this tool, Rescore Plus, implemented in our suite of program VegaZ. And then we used this set of scoring function to generate the linear combination, the consensus models, the linear combination, again by using VegaZ as a platform and using the enrichment factor optimization algorithm. In particular, we developed linear combination including at least three variables, three descriptors for each model. In this way, we obtained quite interesting result for the three analyzed pockets. You can see in this graph the enrichment factor, the best enrichment factor obtained, and interesting result showed that in general the mean value provided better result than the best value, and more or less the mean score allowed to generate a consensus model which provide similar performances in all three pockets, around 16, 18, 16, 15 as enrichment factor. And with this preliminary model, we are able to validate our computational strategies. We are able to better understand the protein and to study the potential of each pocket in order to perform following with a screening campaign when we will have the new experimental data. Next slide. Now we can start with the experimental part, and in particular in the Fraunhofer Center of Hamburg, these assays were developed to measure the unwind activity. In the case, a double strand of DNA labeled was used, and the helicase unwind the double strand, and this unwinding induces a decrease on the fluorescence that can be measured, for example, by suramine and these no synthetic inhibitors. Of course, in order to optimize this assay, were added the ATP and also a second strand, which is complementary to the strand used, the double strand, in order to avoid the rebinding of the two strands and to measure the correct inhibition activity of our tested compound. Similarly, there is also a second assay which measures the ATPase activity by measuring the amount of ADP using this GLOMAX assay, which is a luciferase-based assay. By measuring the
time to wrap up, please. You've got 1 minute left. Thanks.
Oh, then you can shift to the next slide. Sorry. By this now, the idea was to screen the pilot and then the entire European chemical biology library. And until now, the pilot was completely screened, and the full library, more or less, we are to the 30% of the screening. And focusing on the pilot, we obtained 1% of compounds with an inhibition greater than 25 and 0.3% with an inhibition greater than 50%. Next slide. To finish, I'm sorry that is not very clear. However, next slide. No, previous slide. However, even though I don't know why the slide is not so clear, however, we report an example of 10 compound with activity greater than 25%. And the problem that I mentioned before are that some compounds are active on the helicase, but in fact they are no intercalator of DNA, and so they are false positive. And some compounds are redox sensitive. As a matter of fact, only 3 compounds show antiviral activity in cytopathic assay. And so next slide, just to conclude. we can say that the problems of helicase are several problems that render this enzyme a challenging target. For example, the redox sensitivity, the allosteric binding site, intercalation. And so we can hope that the result from the big effort on screening the entire European chemical biology library can provide better result because they can explore new chemotype, they can offer new data for elucidating the emission mechanism, understanding the role of allosteric cavity, and developing improved predictive models. Just the last slide for the acknowledgements, if there is. Yes, I would like to thank for my lab the colleague Alessandro Pedretti and the PhD student Sabato and Pozzi, and from offer Andrea Zagliani and Maria Kurzikov. Thank you for your attention and sorry for my delay.
We perhaps have time for one quick question if there is one. And if not, given the time, we'll pass. Thank you very much for your talk, and we're going to pass straight over to Matthias Tenbusch.
Thank you again, and sorry for my...
Hello? Yeah, thanks for the invitation and for the big support from ISIDORE for this project. And I will talk about mucosal immunity against SARS-CoV-2. Okay. Yeah, so we have all witnessed the induction of new vaccines against SARS-CoV-2 during the pandemic, and we also have seen that there was broadly protective against infection and severe disease in the beginning. But then we also faced the limitation of this systemic immunization via intramuscular immunization, because a lot of breakthrough infection occurred, probably due to waning immunity and also to the emergence of new variants. And thereby also the vaccinated individuals were part of the transmission chains. And in contrast, mucosal immunizations via spray immunization in the nose, for example, are aiming to induce local immunity and thereby prevent the infection directly at the entry place. And thereby it should be also lead to reduced rates of transmission. However, in our previous studies on other respiratory viruses, we already have seen that most probably the best is to combine both arms, and thereby we established very efficient systemic prime mucosal boost vaccinations. And that was also in the beginning of the pandemic, we utilized mRNA vaccines or DNA vaccines, which encodes the spike protein as an intramuscular prime, and that was followed by an intranasal boost with an adenoviral vector encoding the same spike protein. And we compared it in the beginning to the licensed vaccine procedures, so 2 intramuscular injections, either with mRNA or with adenoviral vector 5. And then we constantly checked for the immunogenicity in the local compartments and the systemic compartments. And just to highlight the most obvious benefits of the intranasal immunization is the local antibody responses. So when we analyzed the antibody responses in the bile fluid, so the washing of the lungs, so we can clearly detect IgA responses only in the groups where the adenoviral vectors have been applied intranasally and not in the 2 intramuscularly immunized groups. And that was also translated in better neutralizing activity against the variants of concern which were present at this time of the pandemic. And you see it was completely absent in the 2 systemic immunized groups. And the second part is the local T cell response. So here we have to differentiate between circulating memory T cells and tissue-resident memory T cells. And those cells are specific because they stay in the tissue where they have been induced in the beginning, so they can act directly locally in case of a reinfection with SARS-CoV-2. And to sum that up, you can see this population, again, only in the groups which have been vaccinated intranasally, although you have a large number of systemically induced circulating memory T cells after mRNA vaccinations. But those tissue-resident memory cells have been shown also in other respiratory viruses that they can be more efficient in controlling early viral replication and thereby also reduce peak viremia. And lastly, so we also have shown that those groups were efficiently protected, and which is maybe important for regulatory aspects, that it was a non-inferior protection compared to the licensed vaccine regimens. However, to really improve or optimize mucosal vaccination strategies, we have to understand better what is the best way to induce mucosal immunity and get also durable, broadly protective immune responses. And this was then in the ISIDORE call, where it was targeted vaccination strategies for local tissue immunity. We teamed up with the Primate Center in the Netherlands, the BPRC, which provided models for immunogenicity and efficacy for SARS-CoV-2. And then the BRO promise, so the broadly protective mucosal immunity against SARS-CoV, was born. And here we designed a very expensive non-human primate study, and therefore very much thanks for the support here. And we addressed the boost, in this case, in previously mRNA vaccinated rhesus monkeys, and we have 3 experimental groups. So all of them received twice intramuscularly the Comirnaty vaccine. And then we made a mucosal boost in the form of an oropharyngeal spray immunization with adenoviral vectors encoding spike and nucleoprotein. It's important that those spike is still from the Wuhan. Then we mimicked the hybrid immunity, which was also present in the population. So here we had a natural infection with Delta, or we used a live attenuated vaccine, which is also have been shown to induce mucosal immunity in smaller animal models, which have been also applied by the oropharyngeal spray immunization. And then half a year later, we challenged all of these groups with the XBB 1.5, where we have seen that there is more or less no or low cross-neutralization activity from all these antibodies induced here, and then we characterized immune responses and efficacy. And the first thing, we have to show that there was a clear induction by the mRNA of spike-specific antibody responses, and this is in all groups visible. So we have a peak 2 weeks after the second mRNA vaccination, which then declines to the level, or declines until we perform the boost. And what we can also see is that when you have a look on the anti-spike specific T-cell responses in blood, that there is more individual scatter on the T-cell side than on the antibody response. Because at this time point, all animals have been vaccinated in the same manner. Then we have to make sure that our Delta breakthrough infection was really a breakthrough infection, and thereby we detected a viral RNA in the nose, in the throat, and the bile fluid. And you can nicely see that all of the monkeys show viral loads in the upper and in the lower respiratory tract. So the infection works, and it was really a breakthrough infection. In contrast to that, we only had sporadic infections and viral loads detected in the upper respiratory tract after giving live attenuated vaccines. So that might be the first hint that the pre-existing immunity after mRNA vaccine renders the LAV a little bit inefficient. And that was also seen on subgenomic RNA level, which indicates more or less infected cells. And here you clearly see that there is more or less no infection taking place after the LAV, but robust infection after the breakthrough infection. Then we checked for the consequences of the boost. So we have anti-spike antibody responses in serum, so we clearly see an increase in the antibody response after the boost in the 2 groups which have received the adenoviral vectors or the Delta breakthrough infection, but nearly no improvement by the LAV, what was expected after the viral RNA loads. And most importantly, we also see the induction of IgA responses in the first 2 groups. And for us, more importantly, was the antibody response at the mucosal side. So we took nasal washes and bile fluid again. And again, our experimental vaccination with the oropharyngeal spray immunization with the adenoviral vectors induced very nicely IgA responses in the nose. And also both groups, so the adenoviral vector immunized animals and the breakthrough infection induced IgA in the bile fluid. But there was no local immunity again in group 3. So for the T-cell responses, it was interesting to see. So we have robust spike-specific T-cell responses after the mRNA, but there was more or less no boost effect in all groups after the boost after the mucosal boost. And here we saw some sporadic nucleoprotein-specific responses in the adenoviral vector group and also in the Delta group. And this is because this was a primary antigen contact with a nucleoprotein, so this is a primary response. More importantly, again, the mucosal T-cell response, and here we really had a dramatic increase in the breakthrough infections, so it was up to 40% of the CD4 cells were spike-specific in the Delta group, and we also saw this robust induction of CD8 T-cells. But then after the peak, 2 weeks after the boost, it rapidly declines. And in the adenoviral vector group, we had some different kinetics. So we have still CD4 and CD8 T-cell responses at the time of the final challenge. And again, we do not have any mucosal T-cell responses induced by the mRNA or by the live attenuated vaccine. And the same was true for the nucleoprotein. Again, this sharp increase in the delta breakthrough group. And then you can already see that we have slightly different kinetics here in the adenoviral vector vaccine groups and robust T-cell responses at the time of the challenge. Which brings me to the efficacy of these and the most important and interesting data, which have been finished last week. So the analysis is still ongoing, but these are the preliminary data. And here we again took nasal swabs, so protection in the upper respiratory tract, so genomic RNA, subgenomic RNA detected. And what you can nicely see is in the non-vaccinated group, so you have occurring viral replication until day 4, subgenomic and genomic, and 5 out of 6 animals showed detectable RNA levels. In contrast, so the adenoviral vector immunized animals, so we had 1 animal out of 6 which had sporadic RNA levels detected on the genomic side on day 3, but none of these animals had any subgenomic RNA detected in the nasal swabs, so 0 out of 6. The delta breakthrough infection was intermediate efficient, so here we had half of the animals which showed viral RNA levels. However, they also had lower RNA levels and decreased much faster. And still the mRNA or the LAV group obviously have some protective efficacy in clearing the virus more rapidly. So that might be an indication that they have less severe disease if you would like to translate it to humans. And then in the throat, it looks a bit different. So here the RNA detection was more robust in all of the groups. But again, although you have RNA levels in all treated animals, you see a clear decrease in the subgenomic RNA levels in the first 2 groups, and there was no difference between the LAV group and the non-vaccinated. So this also indicates that mRNA alone, or in combination with the LAV, the one we have used, do not provide any protection against this XBB 1.5 challenge. And then in the lower respiratory tract, we saw again the same picture. The 2 groups which have nice mucosal antibody and T-cell responses were completely protected against RNA or against the viral challenge, and there was more or less no protection against the other ones. This brings me to the conclusion. So the lessons we have learned from this project so far, that the intramuscular mRNA immunization does not induce mucosal immunity, that was shown before, that it does not provide sterile immunity to the VOCs, and perhaps it renders the homologous LAVs more or less inefficient. The hybrid immunity after the breakthrough infection was characterized by a fast and strong mucosal T-cell responses and efficiently protects against lower respiratory tract infection with the XBB 1.5. And our tonsillar spray application of the adenoviral vector vaccine as mucosal boost induced robust mucosal T-cell responses. We had strong systemic mucosal IgA responses, which then translate into the protective efficacy against upper and lower respiratory infection with the variant XBB 1.5. And with that, I would like to acknowledge the people from my group, which have been more or less involved in the mice experiment, and then the big support from the Biomedical Primate Center here, especially Gerrit and Petra did a lot of the work, and then of course ISIDORE for the funding and you for your attention.
Thank you very much. Questions?
No, just a comment, and I'm sure I think William may have said this to you already. I think it's a fantastic project, but you're at a place now that you really need to sit down with regulatory experts and maybe start working on a target product profile to identify gaps and what are those next must-have assays. So I would really strongly encourage you, I think William may have said it already, that ISIDORE can offer you that now, very fast track, just to sit down with one of our experts and to work out what the trajectory should be now. And that would be very beneficial if you go after VC funding next, or if it's after an academic style funding call. But I think it would be a nice added value to this great work. So very well done, nice work.
Yeah, thank you very much. So I'm happy to discuss that.
Yes, coming back to your results, thank you very much. They are beautiful. I just have a question. So I'm totally convinced about the efficiency, but another point is also the long-lasting response. Do you have any idea, or do you plan to test whether the two shots, the hybrid and adenoviral local immunity induction, leads to better coverage during a longer period of time?
Yeah, I think it was already half a year, so we followed up on that, so which is rather a long time for the non-human primate study. So we did some comparable experiment in mice, so where we can follow the immune response longer, but of course you cannot check for the efficacy. But there we can see that our RNA prime mucosal boost at least induced for 1.5 years very robust mucosal T-cell responses. And thereby we think that might also translate in long-lasting protection, but it's not the right model to really test for efficacy. And then for non-human primates, it's hard to do such long studies, I guess.
Okay, thank you very much. So after having heard about inhibitors for the ribonuclease, we've now got the protease inhibitors, Duc du Vau. And just a reminder from the AV team, do look towards the camera, please.
Hello. Hello everyone, my name is Dri, a team member in Professor Jane Garson's group at the Department of Cell Molecular Biology at Uppsala University. I'm very pleasure to be invited here to talk about our project. So we are a group of computational chemists and chemists try to contribute something to the pandemic. So I'm going to present our project about develop novel structure inhibitor of SARS-CoV-2 main protease with broad spectrum activity, and as you may know all about the life cycle of SARS-CoV-2 starting from coronavirus and try to through many steps, and our target is located here, which as the NSB5, the main protease, which has an auto-processing that's cleaved many polyprotein to produce many other protein, participate in the continuation of the formation of virion, and then continue to formation of the viral particle and infect other cell. And this protease has been shown to clinical effective. As you may know, the Paxlovid drug produced by Pfizer that has really clinical impact in human. And so in our project, so we use computational approach. So we use a crystal structure that is available at the beginning of 2020, and also a fragment crystal structure from COVID-Immune Stock Consortium to make a virtual screening of more than 200 million compounds using DOS7, and also a fragment growing strategy of 2 million compounds in a chemical space of more than 10 billion compounds. And then we selected, in which strategy we selected 100 candidates to be synthesized and tested using different in vitro chemical in vitro assay like sulfaplasmin resonance binding assay, enzymatic assay, is co-crystal structure of the compound with the protein, and cell-based assay to develop the novel structure. And so, as you can see here, I can show some of the initial compound that has a quick binding and quick activity with the protein, the main protease, and we also co-crystallize this compound with the protein to confirm the binding mode that we predicted is really matching or similar to the co-crystal one, and that also guide the optimization that I can show you here. We go from some micromolar activity to submicromolar activity, and then when we added the chloro here, that we improve the compound to low nanomolar activity. And the work has been published, and we also confirmed the antiviral activity and broad spectrum activity of this compound by testing in SARS-CoV-2 infected model, SARS-CoV-1, and MERS. In the next step, we try to optimize the activity of the obtained compound to improve the ADME property. In collaboration with different facilities in Sweden, so in total, we have synthesized for more than 400 compounds, testing the more than 200 compounds, and in cell 80 compounds, and also in vitro ADME of more than 100 compounds. And so this is, I'm just a contributor to the whole project, but there's a lot of people involving in at the beginning of project. And then we wanted to profile our compound to show that they are really promising or not. So we need the help of ISIDORE. So ISIDORE provides two survey project that can help us the screening of 60 compounds in different SARS-CoV-2, SARS-CoV-1, and MERS in calustri cell with the help of the group of Dr. Bat, and the evaluation of the best inhibitor in the more cell relevant models, air liquid interface with the group of Dr. Fabien at Lyon University. And so the result of the screening on calustri cell is showing here that you can see of 60 compounds, 40 compounds show broad spectrum activity at micromolar or submicromolar activity, which then we select 5 compounds for testing in the next step in comparison with the approved drug, nimatrelvir, and the clinical phase 3 NC-trelvir. And the reason we selected 5 compounds is because we also consider a lot of parameters in the development of the drug. It's not only the efficacy in different cells, but also the binding affinity, the enzymatic activity, but also stability in mouse liver microsome, human liver microsome, thermodynamic kinetic solubility and thermodynamic solubility. And so, as you can see here in green, that this first 5 compound has a good stability or solubility that we can bring the compound to the next step. And so we tested this 5 compound and compared to the 2 reference compound in the air liquid interface using 2 complementary methods, the quantification of viral RNA and the quantification of infectious viral titer. In case you can see here that the 3 compounds behave similar to the 2 reference compounds, nimatrelvir and NC-trelvir, and we select 2 compounds to go for the next step for testing in different EU strain and Omicron, SP, and BA. And you can see here our compound, what we have, why good compared to the reference compound nimatrelvir and NC-trelvir at 0.1 micromolar or 1 micromolar. It's a bit less in the Omicron. Activity is a bit less in Omicron strain, but it's still significant and comparable to the clinical phase 3 NC-trelvir. And for the activity about the produced viral particle here, in the EU strain, our compound is strikingly also active, as good as the reference compound, and it's a bit less active in the Omicron strain, but for the 285 is still comparable to the clinical phase 3 compound. So in conclusion, during the lead optimization phase, we've synthesized more than 400 compounds and having 80 compounds with good activity in viral E6, and from the screening, the help of ISIDORE, we screened 60 compounds that lead to 20 compounds with very good activity and broad spectrum activity against SARS-CoV-2, SARS-CoV-1, and MERS. And then 5 compounds were profiled against air liquid interface cell that lead to 2 candidates that can be further evaluated in vivo. So I would like to thank the support from ISIDORE and the 2 team that helped us to carry out, and the funding from UK ICN. Thank you very much.
Thank you. Are there some questions? Martin.
Yeah, so in your study you compared your compounds to 2 reference compounds.
Yes.
And you showed them to be as good as the reference compounds.
Yes.
But do you intend to make your final compounds better than the reference ones? Because so far I don't see equal, but...
Yes. So the advantages of our compound is smaller, and it also has a brain penetrant property that the other 2 compounds don't have, but I haven't shown here. And we actually partner with a pharma in the US that actually develop compound further.
Okay, thanks.
Any other questions? Well, that sounds very encouraging. Let's hope that does move forward. It would be a very positive outcome. Thank you very much. Charlotte Primach will be talking about immune responses induced by OVX033.
So thank you very much for giving me the opportunity to present this data. It's based on immunogenicity, cross-reactivity, and polyfunctionality of immune responses induced by the vaccine developed by Osivax, with and without adjuvants, and in an NHP model that are naive or convalescent for SARS-CoV-2. Commence bien. I don't know. It doesn't work. Okay. So Osivax is developing broad-spectrum vaccines against constantly mutating pathogens, and the aim is to provide universal protection against current and future strains of influenza, along with existing and emerging coronavirus infections. So we are focused on providing proof of concept against this, for these indications at the clinical level. And the vaccines developed by Osivax are based on 2 pillars. So the first one is the choice of antigen. So as you know, conventional approach targets surface antigen of pathogens that are evolving and mutating. So the sterile immunity is based on neutralizing antibodies. And because of these mutations, the vaccines need to be adjusted regularly. Whereas Osivax universal approach is to target internal antigen that are less sensitive to mutation and highly conserved between strains. And the second pillar for these vaccines, it's the oligodome technology. So it's a self-assembling nanoparticles. So the vaccines developed by Osivax against influenza and SARS-CoV-2, it's composed of the full-length nucleocapsid for sarcoviruses, nucleocapsid protein from SARS-CoV-2 Wuhan strain that is genetically fused to the self-assembling protein sequence and with T-cell activation features. And when it's expressed on Escherichia coli, this individual elements spontaneously auto-assemble in nanoparticle form. And this repetitive pattern is key for immunity, and this vaccine is able to trigger strong B-cell responses as well as T-cell responses. We were wondering if our vaccine could be associated with adjuvants, and thanks to the network of Transvax and ISIDORE, we start to work with Vaccine Formulation Institute in Switzerland to select a potent adjuvant, and we have identified SQ adjuvant, which is a squalane in water emulsion that contains cholesterol and QS21 saponin. It's a stable emulsion that is ready to be formulated with an antigen, and that enhances antibody and T-cell response, and that skews the immune response to Th1 response. And with the grant from ISIDORE, VFI was able to produce a technical batch for toxicology studies that was successfully manufactured, and now the process is ready for GMP production. So we evaluate our vaccine and alone or in combination with the SQ adjuvant for the efficacy of protection in the hamster model. So the animals were immunized twice, then intranasally challenged with one of those three variants of SARS-CoV-2, and the results were very promising and highlighted a good protection as assessed by less body weight loss for vaccinated animals. The viral load in lungs that was also reduced compared to non-vaccinated animals, and the reduction of lung histopathological lesions. However, there was no significant difference when the vaccine was used alone or in association with SQ adjuvant. So we were wondering if in humans this adjuvant could have an interest or impact, and therefore we wanted to evaluate this vaccine and formulations in a more relevant animal model. So we have conducted this immunogenicity study in non-human primates in the platform of infectious diseases models for innovative therapies, CEA in France. And the protocol was as followed. So 3 groups of naive animals were immunized 3 times, 1 month apart, with our vaccine alone, the vaccine with the adjuvant, or a placebo. And we had access to a group of animals that were primed or pre-infected with SARS-CoV-2 Omicron variant. And these animals were immunized only 1 time with our vaccine alone. So regular blood samples were collected to isolate serum and PBMCs to evaluate the immune responses. And we also have bronchoalveolar lavage to monitor the immunity in lungs. So the aim was to compare our vaccine alone with SQ adjuvanted one, and the vaccine alone in naive animals with the vaccine in pre-infected animals. So first, regarding the humoral immune response, what we can see here, you have the immunization, that we have regular increase of the level of N-specific IgG antibodies in serum with immunization. And in the group of convalescent animal, only 1 immunization with our vaccine alone is able to recall the memory response that was not detected before, but with a level of antibodies that is similar in the group of naive animals after 3 immunizations. We have an increase of antibody level with SQ adjuvant, even if we don't have any statistical difference here. Then we monitor the interferon gamma T-cell response by ELISpot in PBMC. And in that case, in naive animals immunized with our vaccine only, we were not able to detect any T-cell response, whereas using the SQ adjuvant, we have this nice T-cell response at systematic level, at least in 5 animals over 6. And in convalescent animals, very interestingly, our vaccine, non-adjuvanted, is able to have this also this recall or memory response that was slightly detected at baseline. So that was very interesting to see how the fact that the animals were primed is really important for the activity of our vaccine. Then we wanted to evaluate the cross-reactivity of this T-cell response because N antigen, as I told you, is highly conserved between different strains of SARS-CoV-2, but also with other coronaviruses. And here the PBMCs were stimulated with N-peptide pools from Wuhan, Delta, and Omicron variant. And we have really similar T-cell interferon gamma immune response in those cases. So yes, the cross-reactivity of this T-cell response is preserved. Finally, to evaluate the polyfunctionality of this T-cell response, intracellular cytokine staining was conducted in PBMCs first. And what we have shown is that the T-cell immune response was mainly driven by CD4 T-cells because no response was detected for CD8 T-cells except very low interferon gamma response. The response was Th1 T-cell response mainly. No Th2 response was detected as no IL-13 was measured. And we have a nice response in the group of adjuvanted vaccine as well in convalescent animals. Very interestingly too, the SQ adjuvant really promotes IL-17 response that is really not detected when the vaccine is used alone. And finally, the same analyses were conducted in BALS, where the cells were purified and also evaluated by ICS and flow cytometry. And we really have a similar response than in a systemic level. So basically, Th1-oriented CD4 T cells response. Mainly in the group with the adjuvant, but also convalescent. And also we still have this IL-17 response when the vaccine is associated with the SQ adjuvant. So regarding the N-specific humoral immunogenicity, our vaccine triggers strong B-cell response with and without adjuvant in naive and pre-infected animals, and a single immunization of our vaccine alone in pre-infected animals leads to a high immune response. And regarding the N-specific cellular immunogenicity, the T-cell response is detected in animals that received the adjuvanted vaccine, but also in convalescent primates, that were immunized with our vaccine alone. These responses are oriented toward CD4 T cells, it was polyfunctional, and Th1 response, and also this IL-17 response that is associated with the vaccine. And it was cross-reactive T cells and against the nucleocapsid from 3 variants. And this vaccine is currently on a first-in-human trial. So we really wait for data about safety, of course, and immunogenicity in, of course, primed individuals. Thank you.
Thank you very much. So you said that the SQ had an effect in the non-human primates. Has this been seen with other vaccine candidates as well? That SQ is only showing that beneficial effect?
No, it has been shown in other models. We have, with our vaccine, in combination with our vaccines, we have evaluated in hamsters and mice. And in hamster, we didn't highlight such an improvement of immune response, but maybe because we were already at the top, maybe. And for instance, in mice, we were able to show that we can reduce the amount on vaccine and preserve the level of response. So, yeah, we can highlight if we reduce the quantity of vaccine.
Thank you for the clarification. Question?
Interesting presentation. So you mainly have N-specific antibody responses and CD4 mechanism of protection.
Yeah, it's to neutralize the vaccine, the virus. That it's disease-modifying vaccine recognized by the immunity because are expressing the nucleocapsid at the surface of the cells. So it's based.
Mabel?
It shows that your molecule induces a response that is quite similar to what is shown in recovering. One difference seems to be TNF alpha, which is. Do you think it might be beneficial to the response?
I won't be able to answer that question. I'm really sorry, but here it's that level of knowledge. I'm sorry.
Mentioned that the preclinical development of the adjuvanted vaccine is still work in progress. Efficacy studies.
So to evaluate, maybe also we should look at how dose of vaccine, because in hamster we couldn't discriminate the vaccine. And in parallel, VI is developing GMP process, as I said, toxicological studies for safety. This adjuvant was not used yet in clinics, and then maybe yes to go to clinics with this adjuvant. It will also depend on what we will see with our vaccine alone in this first-in-human clinical trial to see if the vaccine is able really to have this recall of pre-immunity in subjects, or if we should increase the level for immune response.
And for the preclinical, for the efficacy study, you would stick to the hamster model then?
It's a easier one and cheaper, of course. So it also depends on that. What we want to pursue now at preclinical stage, it's to evaluate the broad spectra of protection, so going with SARS-CoV-1 or MERS, and to see in that case if the adjuvant maybe, because with this increased immunity, is able to better protect with, from pathogens that are not exactly fit with the vaccine.
Okay. Thank you very much indeed. Thank you. So we're now going to hear about the Swiss surveillance system. And you don't look like Jonathan, so I guess this is going to be a duo between Jonathan Sobel and
Hélène Allegra.
Hélène, yeah.
Okay, so hi everyone. First of all, thanks a lot for allowing us to present our project. We will escape a bit the world of fundamental research. Now it's more closer to the third goal about protecting people. And basically about a year ago, I'm coming from University of Geneva and the University Hospital of Geneva, and I was part of CHSUR, so it's the National Surveillance System for COVID-19 and influenza. It's a targeted surveillance system, for instance, the wastewater that was evoked earlier on. So basically, our interest was to understand a bit more about long COVID, because long COVID is supposedly targeting about 65 million of people today, and it's really an emerging global health issue. So just about the CHSUR system, as I said, it's only in the hospital, and we collect a lot of information about the patients, for instance, the demography, the outcome, the clinical course, and so on. And we had, at the peak of the pandemic, about 21 hospitals that were covering the whole Switzerland. And we use an electronic case report system, which is RedCap based, which is maintained by the University Hospital in Geneva. And this was a mandate of the Federal Office of Public Health, so the Swiss Federal Office of Public Health. And we did a lot of reporting for them, so monthly and weekly reports. We reported back to the hospital themselves, and we did as well some communication for media. So we had often some requests of the media to give like trends and basically different aspects of the new variants. Side to that, we did as well some research. So for instance, we had studies over readmissions or evolution of the case fatality rate and others. But this system is quite limited. As I said, it's only focusing on patients in the hospital, and we do not collect everything for those patients. We do not collect vital signs, for instance, or some lab tests. And actually, the most fundamental limitation for me is that we were not aware about the patient's struggles and needs. So really, it was kind of the medical aspects, but how people are coping with their disease is really unknown. So when I applied to the call of Global Sonar, I had basically this question in mind. I wanted to maybe develop like a large platform that could collect data from outside of the hospital, maybe transnational, and so on. And I was wondering, okay, what should we collect? How can we best empower patients? What information should we provide them back? So what statistics or technical or legal information? And is there any limitation that we need to take into account? And just briefly, we started the project with some literature analysis. And for that, in my previous group in Geneva, we developed a tool called LightRev. And it's a tool that allows to speed up the systematic literature review process. And very briefly, basically, you identify a corpus of paper of interest, And then you compare all those papers by transforming the abstract, for instance, into Word to VEC. And from that, we can compute this landscape that you see here. And inside of this landscape of your topic of interest, you can identify sub-cluster like that, which represent subtopics that are important for your subject. And of course, you can discard irrelevant groups of paper and manually refine at the end your corpus. And starting now, I will give the speech to Ellen, who did the service for me.
Hi, my name is Ellen Algera, and I'm a medical anthropologist, and I don't think there's a lot of social scientists in the room. So anthropology is a field of social science that basically has small-scale but in-depth research into how people live, what they do, and how they give meaning to what they do. And so when Jonathan wanted to know how people with long COVID, what kind of information they need, how they look for information, how they give meaning to their practices, I was, with a group of other people, the service to Jonathan to explore the field, the information field that people living with long COVID are in. And so this is very briefly the timeline. We started the literature search and the ethnography, so that's the in-depth small-scale research we did in November. And we partly did the ethnography online. We looked at how people also exchange online information. I also had a lot of online interviews because people living with long COVID cannot travel, for example, to meet me somewhere. Then in April, we invited a few other researchers to work with us to prepare a questionnaire. So we're also going to do a survey. And then the results of the survey we expect to come in the fall. And by the end of this year, we hope to have 2 papers submitted. One mixed method paper that combines the qualitative data with the quantitative data, and one paper only based on the interview data. So in the ethnography, what we did is we explored the informational ecosystem, including apps that people use and platforms. We interviewed people, about 30 people in these different countries, so the Netherlands, Austria, and Switzerland. And we asked them about what are their practices, what informations are they looking for, are they collecting data, with what tools, and their wishes. And in our analysis, we looked at some of these apps and platforms. We reviewed these platforms, and we did an analysis, qualitative analysis of interviews. We're kind of still in the course of analyzing these interviews. And so what does this complex informational ecosystem look like for people living with long COVID? I think it's important to remember that people who suffer from severe long COVID, this is a really debilitating condition. It means basically people have to say goodbye to the life that they had yesterday and are unable to do the things they were used to do before. So they're not able to work, they're not even able to go outside. Some people cannot sit up on a chair. Some people cannot eat soup with a spoon. Very simple things that we take for granted are impossible for people who have this long COVID in a severe way. And then on top of that, people have to navigate the information to kind of understand what is happening with them and understand how they can manage their illness. And when we interviewed people, they emphasized a lack of scientific understanding of long COVID. So what is it really? A lack of treatments and cures, and also a lack of professional guidance from the care providers that they encounter. And this leads to people leaning a lot on peer support, so online groups, for example, where people help each other. They develop self-management techniques, sometimes on their own, sometimes with an occupational therapist, for example. And people adopt all kinds of new techniques, which was interesting to see. So people use, for example, fitness trackers to help them manage their daily lives. And pacing is really important. So they use these trackers to also not exert themselves too much, because taking a walk may have an effect of having to lay in bed the next day or two. So how long can I walk? People kind of check their wearable, for example, and check their heart rate and heart rate variability to see how much they can do without having to pay the price of sheer exhaustion the next day. So we had several questions when entering the field. We wanted to know how people navigate this complex informational ecosystem, what tools they use, and also what are their wishes and needs for a patient platform. And so, like I said, we found that people are trying to find peer expertise and community among people living with long COVID. They have to do a lot of informational labor, filtering information. What is valuable for me? Is something evidence-based, or is it just a way to make money out of people who are suffering from long COVID? And they also tinker a lot. So they try to find out with the technologies and tools they have, what are my triggers, what helps me recover from certain things. So that was interesting to see. Now people use lots of online sources, high and low tech tools. So some people just use a pen and paper diary to write down the things they do and how they feel. And some people adopt these more advanced apps as a way to manage their lives. And then to come to Jonathan's question, could we develop a platform? The interesting thing was that most people would imagine a platform when we asked this question, but they also had a lot of disclaimers. For example, we already have a lot of patient platforms, so we don't need another one. Or we actually need something else. We need awareness, also in the political field, that there's more research and that there's better care for people like us, that there's better financial support for people who lose their livelihoods. So no platform may be an answer. And then sometimes people said, we want a platform, but we want a platform for researchers or care providers so that they can collaborate and they can make the knowledge, and so that it's not us having to do the work, but it's the people in the research institutes. And then when they imagined the platform, they did talk about questions of accessibility and ability. So for example, screen time is impossible for some time, or very limited for some people. So maybe audio would be a way to inform people. They want tailored information, so information that is relevant for them in their situation. They want to meet peers, but not just all the peers, but peers that are like them, because they feel like long COVID, it hits different people in different ways. So they want to connect, but not with people who have completely different lives from them. They want evidence-based information, and of course, especially how to manage this disease in an evidence-based way. And then the wearable users also wanted to have their wearables validated. And with the survey research, I'm going to go brief with this. We want to substantiate the qualitative findings in the survey. So we're going to send out a survey in the different countries. And there we want to have a representative sample of people living with long COVID. And we hope to get the results in by the fall. Then back to you.
Thank you. So just to summarize what we heard a bit, so what was really surprising to me is that the patients don't want a new platform, really. They are not against like sharing their data, so some wearable data or even some molecular data. They want something that is more integrated with the current initiative or healthcare system. They don't want to have another extra platform. And what I learned from that project overall is that the patient-centric design is very important. So implicating them directly in the co-design will really open the perspective and make something that is more tailored and more wanted by them. As we said, they have different search strategies. They are looking for different types of information, scientific, but not only. They want a better recognition of the disease, and they need some practical advice as well. So that's something that we need to take into account. And that's basically it for this presentation. This is the picture of the whole team. We met in Amsterdam about a month ago. And with that, I would like as well to thank my former supervisor, Professor Olivia Kaiser from the Institute of Global Health, and Dr. Errol Orel, who developed LightRev, and the Federal Office of Public Health, and my current boss. Thank you very much.
Thank you very much indeed. So I imagine there are plenty of questions, but what I suggest is that we move to lunch, and that everybody has the opportunity to talk to you, but also to the other speakers. Before we go to lunch, just once again, thank you to all this morning's speakers, and we're back here at 1:45, I believe, an hour and a half for the posters as well. So please do take the time to visit the posters and discuss with the poster presenters. Thank you very much. Okay, we're going to get started again. I hope you were able to take advantage of that lunch break to look at the posters, discuss science, and we'll get straight back on with now a keynote about risk group 4 pathogens. So I'm delighted to welcome Kirsten Fischer from the FLI. So Kirsten trained as a vet, has a long-standing interest in some very nasty pathogens. So Ebola, Henipa, orthoneurovirus ecology in sub-Saharan Africa and Europe, and trained to work in BSL-4 laboratories. So we're very much looking forward to your talk.
Thank you very much. Thanks for having me. And yeah, I think we've seen a whole bunch of, there it is, different and brilliant talks this morning, and I hope that I can add up to the diversity. So preparedness for risk group 4 pathogens. Let's start with what they actually are. So they have several features in common. They are all viruses, and they all have a single-stranded RNA genome. Yeah. They are either rodent for arenaviruses, bat-borne for Henipa and filoviruses, or tick-borne for CCHFV. And they are all zoonotic viruses. So they have either an animal reservoir or they have an animal as an intermediate host before transmission to humans. They would not be classified as risk group 4 pathogens if they didn't cause severe disease. So some of them may result in hemorrhagic fever in patients. Others can cause severe forms of encephalitis. And depending on the specific virus isolate and or the quality of the healthcare system in a country of emergence, the case fatality rate may be up to 75%. And all of them have in common that medical countermeasures are so far limited. So when we then look at the worldwide or global distribution of these pathogens, we can see that we have New World and Old World arenaviruses. We have filoviruses reported to circulate all over the African continent. Henipa viruses in the Australasian region with some evidence that there are antigenically related Henipa viruses circulating in Africa. And then there's CCHFV, whose distribution greatly depends on its tick reservoir, ticks of the genus Hyaloma. And wherever those ticks are found, there is the chance that they may bring CCHFV with them. So Africa, Asia, and Southern Europe, and the tick has made its way further north already due to global warming. So first reports of the tick have been done in Germany and in Sweden. So that is also one reason why we should be prepared when we come back to the term preparedness. So there's always the risk for emergence of these pathogens, and whenever there's an outbreak or a case reported of these high consequence pathogens, then they continue to cause public health concerns. And in 2015, and because of these limited medical countermeasures, WHO has classified them as priority pathogens, so to intensify research and development to enable improved design of these countermeasures. And I said it, repeated reports of Nipah virus infection also cause public health concern, especially in India. They experience almost annual outbreaks. And then there was the first detection last year in November, I think, of molecular detection of CCHFV in southern France. So there's still more, probably more to come. And when it comes to preparedness, the probably most impressive example of lack of preparedness was the Ebola virus epidemic in West Africa. So there, actually almost nobody really expected the virus to be present in that region of the world. So that gave the virus somehow a head start, and it could silently spread, and that contributed to the unprecedented number of total cases that were registered, and also the highest number of deaths in the history of Ebola virus disease, I would say. And then the question is how? How can we be prepared? And I think with all of these keywords that are probably just a limited number of disciplines from my very limited laboratory-based point of view. They all have to work together, and with each discipline you could fill hours of talks. And for today, I would like to put my main focus on the surveillance and diagnostics part, and would like to show you what we've done at our institute, coming from a Federal Research Institute for Animal Health in this regard. And so we start with Henipa viruses. So in the very beginning, 1990s, there were mostly bat-borne Henipa viruses described, but there's increasing evidence that there is a whole branch of rodent-borne Henipa viruses of zoonotic potential. When you then look at the right side of the slide, you can see the epidemiology of Nipah virus in 2 countries where it emerged, Malaysia and Bangladesh. So either Nipah virus was or is transmitted from bats via pigs as the intermediate host onto humans, or it can be, in the case of Bangladeshi outbreak or Indian outbreaks that are repeatedly reported, be transmitted to humans via contaminated date palm sap, and then human to human transmission is also possible. Here you can see the distribution of the natural reservoir of these viruses. So I mentioned it, fruit bats or flying foxes, of the Pterophidae, it's not my name, so bats. And you can see in the 1990s, the first one was Hendra virus, recorded outbreak in horses and a few humans, and then the Nipah virus outbreaks coming later on. But when you have a look at these bat species, you can see that related bats are also present throughout Africa. And so it was just a matter of time that surveillance surveillance work got first evidence of Henipa virus related RNA in African bats, and also serological evidence was provided in several livestock animals and horses in this continent. And there was even one report on an eventual spillover event into the human population. And so at that point, we were contacted by a colleague that was at that time working in Uganda, taking samples from slaughterhouses from pigs. And she was interested to look at possible Henipa virus antibodies in these pigs. And so we decided to, besides the gold standard of neutralization test for these viruses, develop 2 serological assays, 2 ELISAs based on the one hand on the nucleoprotein that is highly abundant during infection and immunogenic, known to be immunogenic in infected hosts, and the receptor binding attachment protein G, which is also eliciting neutralizing antibodies in infected animals or humans. And so we expressed, first of all, just the Nipah virus nucleoprotein, and we were lucky to collaborate with partners in Canada who had very precious samples from pigs that were either infected with Nipah virus or with Hendra virus, and we saw a good reactivity of the Nipah challenge pig with the antigen, homologous antigen, and also for the Hendra virus, a cross-reactivity for the Hendra virus infected pigs. We then also produced soluble glycoproteins of both viruses, and saw again a good reactivity and cross-reactivity of both antigens, and a similar, let's say, similar limit of detection for both homologous and heterologous infected pig serum samples. And both limit of detection and cross-reactivity was then important for us, as we did not expect to really find evidence for the circulation of Hendra or Nipah virus, but rather for antigenically closely related viruses. And so we were happy to see that at least Hendra and Nipah showed strong cross-reactivity, and we were hopeful to find something in Africa. And that was indeed the case. So we could confirm from a number of pig serum samples that there were, I cut a long story very short here, but that there were indeed antibodies that could be detected by our ELISA testings and confirmed in Western blot reactivity. But when we performed neutralization tests, we did not find virus neutralizing antibodies. But As I said before, we did not expect to really find those viruses circulating there, but rather closely related ones that would elicit cross-reactive antibodies. And so that is also the key message for all the other studies. So they are mainly based on ELISA and Western blot, and only one study had, in our opinion, cross-neutralizing antibodies. And what could be the source of these antibodies is this Ghana virus, a Henipa virus, where there's no virus isolate as of yet, only a full-length sequence, and it remains unknown whether or not this virus has a pathogenic potential or not. So for that, of course, you always need to do, or in our opinion at least, you need to have a virus isolate and test that in other studies. And then the next virus family that I would like to talk about would be the filoviruses. And there are many important zoonotic pathogens in this genus, but I would in this family. But I would like to focus on the ortho-Ebola viruses, and most of these representatives that you can find here are present, described in Africa. Reston virus is one that is known to circulate in Asia, and Bombali virus is the most recent finding. It's actually it's been found and named after the district in Sierra Leone where the virus sequence was discovered. It has been rescued via reverse genetics and is available, but there's no virus isolate coming from an animal host so far. Okay, and that is something I would like to keep you in mind. When we then look at the epidemiology of ortho-Ebola viruses, then it's almost clear that fruit bats are the most likely reservoir, although, as I said, there's still a virus isolate missing from fruit bats. And I've discussed that actually in the lunch break, so it may be due to really the conditions in Africa. It's hot, you never know what's in your sample, if you can preserve infectious virus until you reach the lab. But anyway, so the fact is there's no isolate so far, and humans are an incidental host, and when there is an introduction into the human population, then human-to-human transmission is quite efficient. And now, coming from an animal institute, we were of course interested in the role of livestock. So we've done some pig studies that I didn't bring with me today, but we also wanted to know about the role of possible companion animals, as dogs are running or were running around even during the outbreak, and were sniffing around in medical waste and so on, and are going for hunting into the forest, so maybe exposed even there. So our question was, do dogs play any role there, yes or no? And we collected samples in the aftermath of the West African outbreak, and at that point there was only one study on a possible role of dogs in Ebola virus, in the Ebola virus epidemiology. And shortly after we had sampled all our dogs, there was another study providing first serological evidence of dogs that had antibodies against Ebola viruses in Liberia. Okay, so that's where we started. So again, we used the nucleoprotein of Ebola virus of the Zaire species as our starting point, as our first screening assay, and developed an ELISA with that. We know that, again, with the nucleoprotein, we are not very specific. But we wanted to start a screening with a very broad view. And then we also employed Western blot based on a nucleoprotein and a virus neutralization test, once based on VLPs as a surrogate assay so that we wouldn't need the BSL-4 facility all the time, and then based on an authentic isolate of Ebola virus. And then we developed our workflow and concluded that if we had at least, if we had 3 tests reactive or with neutralizing response, then it was highly likely that these dogs had either an exposure to the Ebola virus of Zaire species, or, because we are not so specific with our antigens, to another ortho-Ebola virus or even Ebola-like viruses. And so we sampled dogs in Moyamba district in Sierra Leone, performed our ELISAs, screened 300 samples, had around about 12% positive, and so detected antibodies against these antigens and could confirm reactivity for a subset in Western blot. But again, nobody at that point said, okay, those dogs had all seen Ebola virus, because as I mentioned in the beginning, so there were other isolates or other viruses possibly circulating that may account for cross-reactivities. And then we performed our 2 neutralization tests, and the first good thing that we found was that actually our VLP assay, our surrogate assay, was correlating quite well with the authentic virus neutralization test, and we found some weakly neutralizing antibodies against the Ebola virus Mainga isolate in the dogs from Sierra Leone, which was a first, so that was new, but of course you have to always consider the titers were not that high. And what is maybe interesting is that a few dogs of our sample collection were even born after the outbreak, so there's no way of having them exposed during an outbreak, during sniffing through medical waste or something. So that is clearly something they may catch while hunting for food in the forests, or other reasons may be possible. And again, very careful interpretation of all our results. So this may, of course, even if you perform virus neutralization tests and you find neutralizing antibodies, it may be cross-reactivity still, because we didn't test a whole bunch of Ebola virus, also Ebola viruses, but only that one. So yeah, that is something we should always keep in mind. And now coming to one of my favorite pathogens at the moment, CCHFV. And when you talk about CCHFV serology, and that's what I would like to do, you have to keep in mind that the genus is quite diverse. You have a lot of different orthoneuroviruses that are known already, and who knows how many are still out there. And when it comes to CCHFV in particular, then there is one virus that is called IGI virus, who is antigenically really closely related to it and could also account for cross-reactivities. So short facts about CCHFV, and I've mentioned that before. So it's at the moment the only BSL-4 pathogen that is really present in Europe and causing, having reports of cases in Spain, the first in France, and there's a lot going on in Turkey at the moment. Also PROMET alerts coming up from Iraq or Pakistan. So it really occurs where the tick vector occurs, and it pushes further north. And now when you look at serology of CCHFV, I like to discriminate 2 periods. One goes until 2018. There we had a lot of different studies from areas worldwide, ranging with a wide variety, of animal species, and all these studies used different methods. So it was a whole bunch of serological methods that you can even think of. Many ELISAs, neutralization test, although that one is not a gold standard for this virus, and indirect immunofluorescence assay coming with a few pitfalls that it's not, at least for animals, not very, very specific. And then there's serology of CCHFV after 2018, and that was when one test was commercialized. And since that, since people can buy this test which has probably a lot of advantages, but it really floods the PubMed with serological studies from really new areas and countries and species that have never been associated with CCHFV before. So, and here's just an example of different studies where people start questioning whether this is real or not. And so here is this test. It's a test from IDVET, and I'm not paid by IDVET. I don't make any advertisement for them. They sell it. It's pre-coded, it's species unspecific, and it is sold with a specificity and sensitivity of almost 100%. But the nucleoprotein, as for the other viruses that I presented to you, is highly conserved. So be aware of cross-reactivities. Then when you really perform this assay, you use 30 microliter of serum per well. So that is pretty a lot. And we recommend testing in duplicate because when we've tested according to manufacturer's instructions, and you only test it once, you be surprised sometimes what is the second testing, what does this look like in another plate. And what is also not so helpful is that the wells come pre-coated, so you cannot test for unspecific background. And then also here we were contacted by partners in Spain, and they were really interested, as it seems in Spain there's a lot in the ecology going on. It's not the classical CCHFV cycle with ticks and ruminants and then transmission to humans. There seem a lot of different animals involved in this ecology, and so they were interested in testing wild boar. And when they contacted us, there were 3 studies published so far, all of them solely based on this commercial test. And so our questions were, are wild boar really possible sentinels? And in that question, we had to consider that for CCHFV, there's actually no real serological gold standard. And when we look at pigs and wild boar especially, there are no defined positive or negative samples for validation, because I think there's no one who has ever infected wild boar in the BSL-4 with this virus. So our study objective was, okay, we would like to develop additional serological tools to really have an idea what's going on in the wild boar population. We had a few samples from Germany, which served as negative controls. We had a few samples from Spain, and we wanted to compare our tests with the ID screen test, and then assess whether or not pigs and wild boar would have neutralizing antibodies. Here you can see where we sampled the pigs and wild boar. So those pigs are really extensively reared pigs, so they have contact to the nature. And the wild boar, of course, they are outside anyway. So we then started with establishing our in-house ELISA, and what we did to account for possibly unspecific staining was we divided the plate in 2 halves. On the one side, we tested in duplicate for nucleoprotein antibodies, and one was mock-coated. We calculated a corrected OD, established a regional cutoff with 334 serum samples from Spain that tested negative in the ID screen ELISA. I admit that this is probably just a proxy to really find negative sera, but it was what we could do at that point, having no other assays set up. And then we tested 518 samples from Spain and found that actually we had a correlation of 67% between our indirect ELISA and the ID screen ELISA, which is not too bad, but could be better, of course. But because we wanted to really know whether or not wild boar pigs could serve as a sentinel, we wanted to further confirm these samples and used specifically, or most of the samples from these ELISA double positive panel. We then, as I mentioned before, established immunofluorescence testings. It was working for almost all of the sample pretty well. And then also performed neutralization tests and found very weak, so those are titers of 1 to 11 and up to 1 to 27. So we say that is really, really weak, but some were neutralizing CCHFV. All of the double positives, almost all except one, were also reactive in the immunofluorescence, and our negative samples were also mostly negative in neutralization tests and immunofluorescence. And then we had some that had discordant results between the 2 tests, and here it seemed, at least from first testings, that that those samples correlated, or the results from these tests correlated more with our in-house ELISA. But we need to do, and that is preliminary, we need to do more testings on those discordant sera, how they behave or not in all our tests. And so when we look at it from an epidemiological point of view, we had no CCHFV RNA detected in any of the serum samples. We had a higher seropositivity in wild boar than in the Iberian pigs. And when we looked at the Iberian pigs, extensively read, those were mostly pigs from one farm. So it could be that at that farm there was something more going on that we don't know yet. Risk factors, that is what the colleagues calculated, was of course, or not of course, but was the age. And female wild boar showed 2 times higher risk of exposure to the virus than males. Okay, so conclusions. For CCHFV serology, I've shown you that we have tried to establish several serological tests in the absence of really defined serum samples, which makes it not so easy. All of them have their pros and cons. The virus neutralization test is, in our hands, less sensitive but more specific. The others are sensitive but less specific. And in our opinion, it is really key for every serological test or every serological surveillance study to combine these tests because they have their limitations. So that is key. We found, but with a critical eye, CCHFV weakly neutralizing antibodies in pigs and wild boar. We don't know what that means at this point. Nobody has ever looked at antibody kinetics in pigs. It may be that nucleoprotein antibodies are longer lasting than neutralizing antibodies. And of course, we have only, as for the Henipa viruses, as for Ebola viruses, tested with one virus isolate and neutralization test, so nobody can exclude at that point that those were cross-reactivities. And once again, for seroepidemiological studies and their interpretation, as I said, confirmatory assays are the key. The virus ecology should be kept in mind, especially in when there are studies from whatever Laos, where there was no report ever of the tick or CCHFV. Then if you have a 99% of seroprevalence in your ruminants, it should not be published, as this also has health implications. And then always be careful with interpretation of serological results. And that goes, and I've mentioned that, with all of these risk group 4 pathogens, as there may be others out there that we don't know yet, which may account for serological cross-reactivities. But surveillance is and stays important, of course, and is one pillar for preparedness. And now just very, very quick, because it's also, of course, something very important if you want to design medical countermeasures of high efficacy. Then, of course, virus-host interactions, pathogenesis is quite important, and all these questions need to be answered to really find points of attack for countermeasures to develop the best medical countermeasures ever. And we've seen a lot of talks about neutralizing or non-neutralizing monoclonal antibodies, and that is also something that was used in the West African outbreak as an emergency use. And we just started to be involved in one ISIDORE funded project where there is a cooperation between Irene Luke from the University of Granada and the FLI, where we would like to use compounds that may counteract the escort pathway for virus spotting, important for virus spotting, and it was shortly mentioned during the talk of Fernando this morning. And those compounds are currently being tested for their efficacy against different risk group 4 pathogens that are named here: Ebola, Marburg, Nipah, and Lassa. And we hope that for the next ISIDORE conference, we'll be able to give you some insight in that. Then please don't read it. It's just to show you that also in the vaccine field of these pathogens, there's a lot going on. It's highly competitive. Some of these candidates have entered phase 1 clinical trials. Some have even been approved for the filoviruses. Highly competitive, really. That's what I what I get from there. And then for CCHFV, there's also a whole list of possible vaccine candidates. At the moment, DNA-based vaccines have entered phase 1. mRNA-based vaccines are also part of different projects that have started now. In one, we are also involved, and that is my last slide. We will, for the next 4 years, try to investigate the suitability of an mRNA vaccine against CCHFV, and we at FLI will contribute with sheep studies and see whether or not an mRNA vaccine may have an effect in CCHFV-infected sheep. And with that, I'm at the end. I thank everybody who was involved, especially our partners from Africa who have always helped tremendously to obtain all the samples, permissions, and so on. And you for your attention, and I'm open for questions.
Thank you very much. Perhaps I could just ask, with the dogs with Ebola, I wasn't quite clear, perhaps you said it, any sign of any disease? Or?
No, absolutely asymptomatic, yeah.
Okay.
Yeah. Mm-hmm.
Yes.
Thank you very much. I really appreciate all the problems regarding specificity. Because, you know, we're working in Africa with many other viruses. We have exactly the same kind of things. And we see really too many papers demonstrating the presence of so-called specific antibodies. Mm-hmm. I think that 90% sometimes are not true. So do you think really that seroneutralization remains a kind of gold standard, specifically, or it's only better than nothing, if I may say? Because you show very good studies, low positivity and very low titers, which is completely different to, for example, against completely different viruses like Zika or many others. Mm-hmm. And for example, what do you think, what's going on in humans infected, especially on the possible decrease over time of specific antibodies demonstrated by neutralizing antibodies on large cohorts? You know.
Absolutely. So, I mean, the biggest problem that we have is that there's no report on when a pig or a dog got infected and raised an immunoresponse, and then, because of that, we can never follow the immunoresponse over time from such an animal. So that is really a huge limitation in veterinary medicine or in epidemiology on animals, where I think for humans at least, you have the report, I was infected, I had a PCR positive result, and then these people can be followed up, and serum neutralization tests can be done to follow the potential possible decrease of neutralizing antibodies.
Even for Ebola, the titers are quite low.
Yes.
So do you think, for example, that seropositive status without any disease exists in animal and humans infected by this kind of virus? This is a major question, no?
Absolutely. It's and I cannot answer it. So I mean, you cannot even really distinguish between exposure or really infection susceptibility unless you do an animal trial with these viruses, I would say. I mean, we could discuss now whether a neutralizing titer of 1 to 45, whatever that means, has to have a real susceptibility, a real infection in front or before. But I would not dare to give a clear answer there.
If I may have a last one. Regarding isolation of viruses, especially in filoviruses, you show, and other people try to do that, like Eric Leroy and many others, nobody has ever isolated the virus from the so-called reservoir for Ebola. But this worked quite well for Marburg.
Yeah.
These are really not the same, but very near, very close.
Yeah.
So why do you think it's like that?
I fully agree. I mean, there are so many studies where they try to find other reservoirs for Ebola viruses, exactly because for Marburg virus it was pretty easy. They went into a cave and then Marburg virus was isolated. Yeah, I think it's questionable whether these Mpox condylorus bats are really, people are trying to force these bats into the reservoir niche. I'm hesitant. I'm really hesitant, but I have no other answer than it's likely, but as long as I do not see a virus isolate, I wouldn't believe in anything. So for me, also the proof would be a virus isolate from bats, and if that is not the case, then we have to sample again and again.
But the European Commission will not allow to kill thousands and thousands.
No.
Slowly by slowly of animals. This will change also.
Yeah.
Okay.
Mm-hmm.
John.
Thanks a lot for a really fascinating talk. ISIDORE is a project with many infrastructures, many of which could be helpful in studies with these really horrible pathogens. Mm-hmm. I was wondering, since you have an ISIDORE project, how much of an obstacle do you perceive that this is? Because I'm representing imaging with 192 facilities around Europe. Very few of them have even BSL-3 labs, not to mention BSL-4. So is there anything we could do to, alleviate any obstacles? And how much of an obstacle do you see that this is?
You mean
for you or anybody working with nasty pathogens of this kind to really obtain this kind of services? Let's say imaging, but there are lots of other services that could be obtained in ISIDORE.
You mean that if we wanted to image our viruses?
If you wanted to image or use any of the other many infrastructure partners that we have in ISIDORE.
I mean, there are a lot of limitations when it comes to sending around samples from a BSL-4 laboratory, so most of them are probably inactivated, and then the question is really what can we measure with that? Of course, there is the possibility to ship around active virus, but we don't like that. I wouldn't honestly see how ISIDORE could help could help us to go around our internal standard operating procedures. So I think those are really higher and make it difficult.
So maybe fixed samples and things like that, safe samples.
And the alternative would be a joint research activity to bring some of the techniques into the BSL-4 labs, which is the capacity, the technical capacity. Do you want to take a microphone?
I was going to say that not in ISIDORE but as Erinia, what we're trying to do is more to bring more techniques into the BSL-4 rather than get the BSL-4 work out of the maximum containment facility. So it's more about upgrading the capacities of the high and maximum containment. I think you probably you have a joint research activity with Erinia actually on the specific diagnostics also and that kind of issues. But yeah, it's not necessarily about going out. It's more about going in.
No, I mean, we're always open and welcome for any ideas that we could bring in. So yeah, whenever there's any.
At Erinia, we've been talking about live imaging like quite a lot and with a lot of stars in our eyes, doing that in maximum containment.
Okay, super.
Okay, in the interest of time, thank you very much. So we're back to a short talk. Lydia Boussicot, who's going to be talking about hemorrhagic fever viruses.
So good afternoon, everybody. First of all, I would like to thank the organizer for the invitation to talk about our work on entitled Targeting Host Cholesterol Metabolism to Inhibit Replication of Highly Pathogenic Hemorrhagic Fever Viruses. So I am Lydie Boussico, and I am Discovery Project Manager in Meletios Therapeutics. So first, just a few words about Meletios Therapeutics. So it's a small biotech that was created in 2020, and our goal is to fulfill the high unmet medical needs regarding current and emerging viral diseases. So we are focusing mainly on RNA viruses, and more particularly on respiratory viral diseases, arboviruses, and hemorrhagic fevers. So how to fight this infection? We are developing disruptive technologies, and we are two main complementary programs. One of them is to develop defective interfering particles, and the other one is to develop broad-spectrum host-targeting agents. To do so, Meletios relies on the seasoned operational teams with strong track record in both big pharma, biotechs, and we also have people skills in preclinical and clinical development. So just to be precise, we are only 6, so it's a really small company. But we also have the support of top-class experts, and we are very lucky to be supported by Felix Ray, that works in the Institute Pasteur, Marco Vinuzzi, and also Vincent Maréchal. So, as I already said, we are developing two complementary programs. The first one is a host targeting agent. So the idea is to prevent the virus from hijacking host resources, and we are focusing on cell host metabolism. So in this program, our most advanced candidate has already shown immunomodulatory activities against respiratory viruses. We have safety data on human, and the next step for this candidate is to have a proof of efficacy in human. We are also developing new candidates, and we would like to develop them in the RG4 application. In our other program, we are developing a platform aiming to develop defective interfering particles. So in that platform, we are designing RNA that will interfere with virus replication, and we are mainly focusing on arboviruses like Zika virus and dengue, and also alpha viruses such as chikungunya. So now let's talk about this project we have with ISIDORE. So why it is interesting to target the host to fight viruses like Ebola. So here you can see that in the literature there is several evidences that virus and Ebola virus is dependent on cholesterol at several steps of its viral cycle. So first, cholesterol and more particularly lipid rafts are involved in, in the attachment of Ebola virion. And it was also shown that cholesterol promotes Ebola virus fusion with the host. Cholesterol is also important in the budding part of the cycle, and it was also shown that the Ebola virus that are budding from cells depleted in cholesterol had reduced infectivity. So in this context, it could be an interesting strategy to target cholesterol metabolism to hamper Ebola viruses to perform its cycle. So, I'm sorry for the for this. So in malicious therapeutics, we are developing first-class candidates with dual activity with both antiviral and immunomodulatory activities. So as you can see here, cholesterol biosynthetic pathway is quite complex and is composed of several enzymatic reactions. So we are mainly interested to develop new inhibitors of enzyme located in the end of the pathway to avoid the side effect of the statins, for example. So as you can see here, statins inhibit the first steps of this pathway, and it could have some impact on isoprenylation pathway, for example, partly explaining their side effects. So by targeting the end of the biosynthetic pathway, we will both regulate cholesterol content, but also intermediary sterols that could have by themselves activities. So that's why we think that we can have activity both on membrane fluidity and also immune signaling. So to design and identify new candidates, we are collaborating with Nostrum Biodiscovery, that is a company that proposes virtual screening and also protein modeling. So in this context, we were very interested by testing our molecule against Ebola and also Nipah virus. But for a company like us that doesn't have a laboratory, it is very difficult to access to BCL4 laboratory. So about 1 year ago, we had applied to the risk group 4 pathogen call from ISIDORE. And that was a very nice opportunity for us to advance in this project. So the aim of our project was to assess our molecule named Totemic-1 activity against Ebola and Nipah viruses in vitro. So for that, I would like to thank the BSL4 laboratory for Jean Mérieux that really helped us for designing the experiment and with who we have a great scientific talk. So this study is the first one of a long project, we hope. So it is pretty simple. First, we choose to work on HUH7 cell line because these cells are metabolically active. That means that the cholesterol biosynthesis pathway is active in these cells, and they are permissive to both Ebola and Nipah viruses. So we have pre-incubated the cells with our molecule for 1 hour prior Ebola infection and collected the medium 48 hours after infection to perform TCID 50. So today we only present the result on Ebola viruses, and the Nipah experiment is still ongoing. So first we have checked the cytotoxicity of our molecule on this cell line, and we observe that we have cytotoxicity, major cytotoxicity appearing around 10 micromolar. We tested, in consequence, the antiviral activity of our molecule and a dose ranging from 30 nanomolar to 3 micromolar. And we observed that our molecule dose-dependently inhibits the virus infectious titer. And this reduction was significant for 1 and 3 micromolar doses. So now, as I said, our molecule activity against Nipah is still being assessed, So these results are really the start of the story. So we will need to confirm the antiviral, but also the immunomodulatory potential of our molecule in more physiological model, in vitro model. So this morning we talk about organoids and this kind of model, but we would be very interesting to perform tests on this model prior to go in vivo testing. It should be also very important to decipher our molecule mechanism of action by performing additional experiments. So for example, we could use pseudoviruses to study really the entry process, but we could also perform cholesterol complementation or kinetics experiments to better understand our mechanism of action. So we would like to really thank ISIDORE and Claudia and Mabel for their help, and also Horizon Europe and the BSL4 lab. And I would like to thank you for your attention.
Thanks very much. Questions? So, would you have done this study without ISIDORE? Would Melitos have carried out work like this had it not been for ISIDORE?
Clearly no, because there are very expensive experiments, so we can't finance alone this experiment. And it is also, we need highly skilled help to really understand how to manipulate these viruses, because they are very special. So yeah, we really need help from diverse collaborators. So if you are interested by working with us, we would be very happy to collaborate.
So this set of results are really promising, but you spoke only about totemic 1, and do you have some means to improve the efficacy, the antiviral activity first? So you can modify, as shows this morning, modify the molecule, or do you have some other molecules such as totemic 2?
That's a very good question, thank you. Yes, in fact, we are working with NBD in Spain to improve the design of our molecule. So yeah, it's a project we have, and it's going on. So maybe we will have new molecules to assess in the future.
Could you use the microphone? Oh, is it not working?
Hello, yeah. So your molecule, it is working as an antiviral and also as an immunomodulator, right?
Yes.
The same molecule?
Yeah.
The same molecule has two properties. That's what you're trying to say here?
Yes. Yeah.
Okay. So have you tried to see any other immunomodulator, like along with this molecule as a combination therapy? Have you taken any other immunomodulator, or do you have planned to? Means I do understand your molecule is working as an immunomodulator and also as an antiviral, but just to see if you can add any other immunomodulator to enhance the potency of your molecule.
You mean
the existing molecule?
Our molecule with an immunomodulator?
Because this is working via the cholesterol biosynthesis pathway. That's why you're telling, isn't it?
You mean with an immunomodulator?
Yeah, immunomodulation part.
Yeah. In fact, we were thinking about to do the other possibility, so combine our molecule that have maybe stronger immunomodulatory activity than antiviral one with an antiviral.
Okay, that's also would be a good approach. Yeah, okay, good.
Thank you very much.
Thank you.
So our next keynote will be from Rémi Charel, who should be online. We can see you, Rémi. You can hear us?
Yes, can you hear me?
Absolutely perfectly. So as you can see, Rémi is in Marseille. So I came from Marseille on Monday, so I fully understand why you've stayed there and not come to Belgium. But we're looking forward to hearing from you. Rémi trained as a PhD MD. He did a postdoc at Galveston, WHO Reference Center for Arbovirus Diseases for a couple of years, then went back to France and has worked between the university and the university hospitals, specifically at the unit of emerging viruses, where he's deputy head, and he's head of the nosocomial infection control department at the Marseille hospitals. Extensive experience in arboviral diseases and very much involved in a variety of EU projects, such as Durable, EVA, and most recently, Evora. Over to you, Rémi.
Can you see the slides correctly?
Yes, we see the old port at the moment.
Okay, and they pass.
Yes.
You see the second slide now.
Yes.
Okay, great. So we'll start. So first of all, I would like to thank the organizer and specifically Audrey for inviting me, and Jonathan as well, to give this lecture. So they didn't give me a very easy topic because arboviruses are so vast. So I had to make choice, and I did some choice. I hope you will enjoy them. So the first idea is to address some general points, and then we're going to talk a little bit about dengue, and then West Nile virus. And to finish, I will present the platform of production of molecular reagent we have developed in Marseille, and that produce lyophilized reagent for diagnostics and detection of viruses, which is a key point of countermeasure and surveillance. So first of all, I think this is the perfect slide to demonstrate what is an arbovirus. So an arbovirus is not taxonomic entity. Arboviruses belongs to very different groups, families, orders, genera, viruses. Some are DNA viruses, few, one only. Most are RNA viruses, but you have single-stranded RNA, double-stranded RNA viruses, negative sense, positive sense, ambisent replication. So basically, they adapt, they are very flexible. So the only common thing they have is they need an arthropod to infect the host, whether it is the reservoir host or a dead-end host, such as human. So as you can see on the left, the number of arthropods that can transmit these viruses are quite diverse as well. So I had to choose from the top to the bottom. You have Aedes albopictus, tiger mosquito. Then you have Culex mosquito, the common mosquito, the one that make your night terrible experience. And then you have this tiny little very nice sandfly gorged with blood, and after you have two ticks, Nixodes ticks, I think, and the last one is a yellow mud tick. So basically, in any case, the infected arthropod, so the arthropod is infected by a virus, it feeds on a susceptible vertebrate. After an incubation period, the vertebrate has a long or short-term viremia, and it can infect through this viremia uninfected arthropods. So it's a cycle between vertebrates and arthropods, and they repeat and repeat and repeat. And here you have the cycle. As you can see, the impact in public health of vector-borne disease is quite important, very high in the intertropical region. But due to the global warming, the intertropical region tends to move north more and more and more. And so basically you will see that the situation has quite changed for the last 20 years. So if we could, this is a list of emerging viruses as listed by the Institute of Medicine. And as you can see, the ones that are shown by arrows are arthropods-borne viruses, arboviruses. And most of these emerging viruses, including the arthropod one, have some common feature, common characteristics. So as I said, in most cases, they have RNA genome. In most cases, they circulate mostly in southern countries. Very frequently, they are either arboviruses or zoonotic viruses. And they are classified for biosafety in biosafety level 3 or biosafety level 4. So basically, they pose some problem of manipulation. So as you can see in this list, which is an old list, you have at least 5 different viruses that are listed by the Institute of Medicine as emerging viruses, and which are arboviruses. So Albopictus is quite the paradigm on invasiveness, but as any paradigm, you can see the forest for the trees. So in French, it's the trees that hide the forest. So basically, is everybody think about mosquito, few people think about ticks, very few people think about sandflies and chilicoides. So, but all of them are quite important in the transmission of these arboviruses. So here you have in yellow viruses that are transmitted by Aedes mosquitoes. So of course you have dengue and Zika virus that belong to the family Flaviviridae. They cause fever and fever with hemorrhagic manifestation. Chikungunya is also transmitted by Aedes mosquito. It's fever with arthralgia. But in the Flaviviridae family, you also have neurotropic viruses such as West Nile, Uzutu virus, and tick-borne encephalitis virus. West Nile and Uzutu transmitted by Culex mosquitoes, and tick-borne encephalitis by ticks, Ixodes ticks. Synbis is transmitted by Aedes and Culex, and belongs to the Togaridae family together with chikungunya. As you can see, you have other families of viruses, including Rift Valley fever transmitted by mosquitoes and Toscana virus transmitted by sandflies. And last, the one that was presented by Kirsten is Crimean Congo hemorrhagic fever virus, which is not only a BSL-4 virus, but also an arbovirus. And that tend to pose some problem to Southern Europe at the moment. But you also have more than 500 other viruses that can cause human disease or can be zoonotic and causing human infection. So basically, this list is strictly non-limitative, and you have to expect that more and more viruses are going to emerge with the time and with the better preparation, because we do diagnostics much better than before. So here you have a kind of summary of how specific can be some viruses with insect. So I will pass on this one. The interesting things is that for a very long time, these viruses were mostly circulating in southern on the Mediterranean shore, and very few cases in the northern Mediterranean. And basically, these tend to be more and more balanced. And now you have the same viruses north and south of the Mediterranean. And even the number of cases that are observed in the southern Europe, so the north shore of the Mediterranean, is increasing in size, in number, in frequency. So the climatic change, I don't know if it's global warming or not global warming, but basically the climatic change for the last 30 years have quite balanced the circulation and the representation of these viruses around the Mediterranean Sea. So let's look a little bit about the mosquito cycle. So mosquito has 4 different stages. The adults lay eggs, become larvae, pupa, and adult again. And when an adult takes the virus from a vertebrate, an amplifying host or a reservoir, he gets infected during his whole life, which is quite short, it's about 3 to 4 weeks maximum. And after the eggs get infected as well, the larva is infected, the pupa as well, and so the adult emerging from the eggs is also infected. So the rate of venereal transmission and transviral transmission is not 100%, but it's something to be taken in consideration for the persistence of these viruses, even without a demonstrated reservoir or amplifying host in the cycle. So as you can see, there are proof and evidence that the cases and the number of infection of some arbovirus infection are tightly associated and links with the climatic change. So this has been proven. The viruses that have the blue dots are the ones that are infected humans and causing human disease. The other one are causing zoonotic disease or veterinary disease with no impact on humans, except malaria, of course, but it's not a virus. So as you can see, the change of climate is playing a huge role in the dissemination of these viruses. And so far, at this time, the tendency is to go from south to north, which may change with the time, but for probably this has been the case for the last 30 years, at least. So let's talk a little bit about dengue. So dengue has been largely increasing since the 70s. This is the reported cases to WHO. Of course, these cases are diagnostic, I mean, proven, confirmed cases by diagnostics. And so they only represent 1% of the total case that claim annually. But as you can see, since the 70s, number of cases or annual case is climbing dramatically. And there is no reason for a change. So the reason for that are multiple. As you know, from 1930 to 1970s, there were very aggressive fighting by spraying insecticide, at least in South America and Central America. And as you can see, the results of this on the panel on the left in green is that the population of Aedes aegypti had dramatically decreased in South Africa during this period. Afterwards, the fight against the mosquito has stopped for many reasons, political, cost, and so on. And as you can see, the situation is exactly the same and even worse than it was about 1 century ago. So in parallel, the number of dengue cases and dengue hemorrhagic fever cases reported in the same region has increased. So basically no case reported in the 1980, and now a quite large number of cases. The third point is the diversity. As you probably know, dengue virus is not a unique virus. You have 4 different viruses. These 4 different viruses are dengue 1, 2, 3, 4. There are 4 different serotypes, and they are not cross-protective. So you can, during your life, get infected with at least 3 of these viruses. Usually, after 3 infections with 3 different dengue virus, you get immunized by cross-protective, because you have cross-reactive antibodies. In any case, they are they tend to limit, and I don't think there is any case of one person having 4 different episodes of dengue during his life, but maybe it can be found somewhere. So the important thing is that in the panel A, what you can see, and the panel A is again the 70s, the serotype distribution was quite stable and limited. Dengue 1 and 2 in the Americas, dengue 1 and 2 in Africa, and the 4 serotypes in Asia. And 40 years later, it's a total mess. Basically, everywhere you have all the serotype of dengue. So it means that the blockade, the reduction of the fighting against the mosquito, but also the increasing number of exchange between continents, they are not human exchange, but also commercial exchange, has dispersed dengue virus, I mean the 4 serotype everywhere. So basically now you can get infected with one of these 4 different viruses in any place of the globe, provided you are in the intertropical region. So the second point will be, we're going to talk about the last emergence of the 20th century. In September 1999, arboviruses for the first time took over from HIV. I love this title. Basically, this was the time when West Nile entered in the United States. Before 1999, West Nile was not present in the Americas. In August 23, 28, 1999, you see that the chief of infectious disease and one hospital recognized neurological disease in several elderly patients. She alerted Marcel Layton, and they both sent samples and spinal fluid to the New York State Department and to the CDC. And less than 1 week later, CDC sent epidemiologists to New York City to investigate what was called at this time encephalitis of unknown etiology or unknown origin in New York City. Late in this Sunday afternoon, 29th of August, in Fort Collins, Colorado, John Roerick received a telephone call from CDC Atlanta. We have 3 human cases of encephalitis in New York. Can you send us some specimens? The meeting was called at CDC first thing on Monday morning. First assumption was an arbovirus, right time of the year, symptoms. The first tests that were performed were ELISA tests looking for IgM, and they used geographic panels. And the results came back positive for flaviviruses as a whole, but also sanguinencephalitis, but with a small titer, I mean little titer. And they were negative for alpha viruses and Bunya viruses. So the message was sent directly to the mayor of New York, and the next day, the mayor of New York, Rudy Giuliani, the famous, announced a 6 million campaign to get rid of mosquito in New York City. And the helicopters start to spray malathion across the city, alarming people much more than the threat of St. encephalitis. So here you have looking where the spraying must be done in New York at this time. So to do that, they use spraying for adult and also for larvae. And it was processed very, very, very rapidly and massively in New York. What is interesting is that basically 1 month later, CDC scientists reported that the mosquito illness was not St. Louis encephalitis, but West Nile virus. And at this time, West Nile virus had never been detected in the Americas. So the tests that were first done by different group of people, Ian Lipkin, Jonathan Smith, and Bob Lanciotti, determined by using maybe for the first time genome sequencing that the disease was not caused by St. Louis encephalitis, but by West Nile virus. The reason for this mistake is that CDC did not include West Nile in the test because it was a geographical panel test. And so in the geographical panel test, you don't include viruses that are not circulating in the geographical area. And so basically what they found is that they detected St. Louis encephalitis, but they didn't detect West Nile virus, which was not included in the panel. So it's very good lesson is that basically in emerging infectious disease, you have to be prepared for the unexpected. So here are the results of the first year with West Nile encephalitis in New York, with almost 10,000 persons infected, 2.6% of the population infected, almost 2,000 febrile cases, 62 cases of encephalitis, and 7 deaths. Horses were also affected by the infection, and also birds were infected. A massive death in crows and birds was observed in the city at the same time. So here you have the progression year after year, because once the virus has entered the US, it never left the US. So this is the first recording of birds infected with West Nile in 1999. In 2000, so basically 3 months later, in 2001, 1 year later, and again 1 year later. So basically, the virus wiped the entire US from the eastern part to the western part very progressively, very methodically, and very efficiently. What is interesting also is that the phylogenetic tree investigating the West Nile genome in the Americas confirmed that the virus was only introduced once. So the single introduction of the virus in this continent has caused a major emergence. Here you have the results that can see global on the top panel. So as you can see, 1999 to 2003, basically the whole United States have been wiped out. So arboviruses can invade territories much larger than Europe very, very fast. And there is basically nothing to do to prevent this and to control and to mitigate this progression. It's the same thing for humans in the panel below, and with the positive counties in pink. Okay, so this was, so as you can see, what happens in terms of progression from 1999 to 2004. So basically, after 3 to 5 years, 4 to 5 years, the entire United States, as well as Canada and Mexico, were invaded with West Nile virus. And this is the same with different data for birds, horse, and humans. So this is the total of human cases during this 7-year period. As you can see, there are neurological disease, but also fever, and also other manifestation. And the peak basically occurred about 4 years after, or 3 years after the introduction in the United States. So what is interesting in this arbovirus also is that their seasonality is tightly associated and linked to the vector seasonality. You don't have the vector, you cannot have the disease, and you cannot have the virus circulating. So what you see here is what was observed in the US. So basically the first case, every year, the first case were observed in June and the last case in November. And this can be applied to many different viruses transmitted by mosquito or even sandflies. Ticks is different. The second thing is now with the, at least in Europe, with the climate change, the period of activity of the mosquitoes is much longer. And the first case of West Nile this year in Spain was recorded in March. So basically the period of activity for West Nile now is from March to November, maybe December. And with the very mild winter that we are experiencing now, it's possible that you will have mosquitoes all around the year. So of course the density is much higher in the warm period, but basically progressively this peak of activity of the mosquito will increase without any hope, I would say. So this is a clear tendency to expand, and we see that every time at the National Reference Laboratory with cases of different viruses transmitted by mosquito or sandfly or culicoides at certain period of the year, even when the surveillance period is not active, it's not launched. So the surveillance period has to be modified and expanded probably to mimic what's really happening in the field. So this is a slide that was produced in 1999. It was about emerging infection and what are the future challenges. As you can see, there are the pandemic flu, antimicrobial resistance, foodborne disease outbreak, urban yellow fever in Latin America, and the unexpected. And you can see that the unexpected is much more frequent and must be expected than it was thought at this time. First, we had the West Nile virus introduction in the Americas. After, we had the SARS-CoV-1 episode and the flu A H1N1 2009, and then the globalization of chikungunya, which was previously restricted to Africa and Asia, but also the globalization of Zika, the SARS-CoV-2, monkeypox now. So basically, the unexpected must be watched because it is becoming quite frequent and it will be more and more frequent. So basically, to tackle this very frequent episode, we must develop tools to be ready and to be able to mitigate and to detect all this emergence as soon as possible. So regarding this point, the molecular diagnostics of emerging viruses are challenging for some reason, and some of them are regulatory reason. And as you may know, for diagnostic kits now, you have to comply with certain regulations of FDA regulation. And in Europe, we have CE-IVD, which will become and has become, already has become, IVDR, which is a regulatory obligation. So the fact is that these regulatory obligations are precluding any rapid response in the field of emerging viral disease. You cannot qualify a kit within 1 month or 2 months from LDT to IVDR. LDT is laboratory developed test. So basically to get the IVDR stamp, you need probably at least 1 year or 2 years for filling the files and the administration and everything. And what happened with SARS-CoV-2 is emblematic, because basically this was the first time that the IVDR or CE-IVD regulation was a problem in the dissemination of the kits and registration in the different countries. So for that, what we developed in Marseille is what we call EVAM, European Virus Archive Marseille, which is a part of the European Virus Archive. And the aim of this platform of production was to increase the level of quality of reagent that could be used for molecular diagnostics, and to increase the quality to ISO 13485, and to be able to distribute this material, and to ensure that this material were qualified and validated for most of the generic real-time PCR kits. So the idea at the beginning was quite simple, is that if you follow the small black square, when you want to do molecular diagnostic in your laboratory and develop a laboratory developed test, a LDT, you need primer, you need probes. They are obtained in liquid format, and you have to accommodate this stock solution, working solution, diluted, frozen, unfrozen, and so on. And so you have many cycles of pipetting and storage at -20 or -40, and everything that must be shipped should be at the cold chain. And we see that in the second pane, the less step, the less mistakes. And so we came up with this idea to mix all of this with the appropriate concentration for optimized results. And if you have a classic RT-PCR assay, it's 2 primer and 1 probe, but in most cases now, the people tend to have 2 different targets, specifically with RNA viruses that can mutate. And so the advantage of having 2 targets in the genome, it's also robustness of the results. So basically you mix everything, you lyophilize this, and you are ready to resuspend this material and to use this together with your RNA or nucleic acid and your enzyme mix. This has been shown as efficient as the liquid formulation in many studies now. And the advantage of this lyophilized primer and probe format is that you have a unique vial, so you suspend your vial only 1 time. When it's lyophilized, your vial is stable at room temperature, so shipping can be done without cold chain. When it's resuspended, it's stable at 4 degrees for at least 1 week after rehydration, and it's basically compatible with all the generic RT-PCR kit, and so you don't have to modify the kit that you use in your laboratory, and you can combine the lyophilized primer and probe with the generic kit that you are using. So this is the second point, and we have shown, and so far we have no example of problem of mixing this, of combining this primer and probe with any brand of real-time RT-PCR kit. The second point is that to ensure quality, which is always the problem of laboratory developed tests, we wanted to use the same criteria for quality as they are in the FDA, the MDCG, and the IVDR compliancy. So basically, there are limit of detection 95, there are clinical phases, preclinical phases, clinical studies. And we have in France something that is called COFRAC rules. Basically, they are the same with different steps to be validated in order to be sure that your test has a good sensitivity, has a good specificity, is not impacted by this or such inhibitors. And what we also wanted is that apart for testing that together with generic real-time PCR, we also wanted to validate this with what is now more and more frequently offered by companies is this open access format in random access automates. And we demonstrated that these assays are adapted to CFADE, to QIAGEN, QIAGEN is the NeuMoDx, Ologix, the Panther-Fusion, and also we have collaboration with Altona Diagnostics to complement their panel of viruses. So here you have a list of 92 assays that are currently available on the EVAM platform. For, as you can see, more than 50% of these viruses are either arboviruses or zoonotic viruses. They have the advantage to be developed, can be developed within, let's say, 1 month with a good certitude of quality. And so it's very rapid response to emerging situation. This EVAM platform is also part of the ISIDORE consortium. So if you are interested, you can contact us, and we can also tailor-made some of this product in specific action promoted by ISIDORE. So basically, I still have 2 slides. This is the take-home message for this EVA platform. So the advantage is an easy access to a large catalog of resources for preparedness and response by using lyophilus products that are well adapted to low-cost shipping to laboratories, specifically laboratory in the south. We can produce inactivated viruses. We can produce primers and probes, and we also produce the positive controls in the form of armored RNA. The formats are tailored, and so basically you don't have any problem, and you don't lose you don't have to I don't know how to formulate this. Basically, it's cost effective because you receive the vial. They have 24 vial reaction, and basically, due to their stability, you are not forced to use them very rapidly. There is a long-term stability through lyophilization. They are adapted to all generic real-time PCR kits and cycler, and they are qualified using all the highest standards for diagnostics. The more global take-home message is to, as you can see at this time, there are at least 10 arboviruses that are currently affecting Europe. There are probably more to come, no doubt about it. Some of them are already endemic. In the case of West Nile, Uzo2 tick-borne encephalitis, and recently Crimean Congo hemorrhagic fever. More are imported, dengue, chikungunya, Zika, but they might become endemic soon, specifically for dengue. The number of epidemics and cluster of cases, whether we are dealing with endemic or imported viruses, in increasing in frequency, in number of cases per episode. So the preparedness and response must accelerate in the fields of diagnostics, but also antivirals and vaccines. And really, the efficient countermeasure must adopt the One Health approach, which means that, as you can see, most of these viruses are zoonotic viruses, so we need veterinary input, environment input, climate models input. Almost last, the EU regulation of diagnostics must also adapt to this new reality to provide validated assay for detection of viruses as soon as possible after the emergence, and despite business plan is questionable is most of the case. When we are talking about sin-based virus, I don't think there is a business plan for a company to develop a sin-based virus test, specifically if this test must be IVDR or FDA approved. There is no money to gain in this. So you need, and we need, to have a parallel pipeline to be able to develop these tests outside of the business plan or the commercial plan. So last point is that, regarding this last point, I strongly believe that ISIDORE is a unique opportunity. And I thank you. This is the same Marseille by night. I hope I was not too long.
Thank you, Remy. Sufficiently long that we have time for one quick question, if there is one. So, Remy, what would you consider to be the most urgent arbovirus threat for Europe?
I would say two. The first one is Crimean Congo, because I think it is currently in Spain, it is in France, it's probably in Italy as well. And I'm not very afraid by the number of cases, but by the fact that it's a viral hemorrhagic fever with high case fatality rate, and that there is some place for panic there. And so I think it's very important to address this, not only through diagnostic or antivirals, but also through sociological perception and sociological studies. I think it's something to be considered very seriously. The second one, it's not because people are afraid of it, except the French government for the Olympic Games, it's dengue virus, I would say, because what we see, at least in France, I would say two of them. I mean, dengue, it's most the problem of France. We have a large number of imported cases, large cluster. Cluster have moved from 3-4 cases to now 20-25 cases at the time. So one imported case can cause 25 cases around just by mosquito biting. So I don't know if there is a question of better competence of the mosquito or higher density or both or something else. But basically dengue is the one that worries me, and the becoming of dengue endemic is also worrying. Of course, West Nile, because it's still a major but it's more the West Nile, more the problem of Italy and Greece at this time. So I would say CCHF and Crimean Congo.
Okay, thank you very much. We're going to pass now to short talks again. So we have Pablo Suarez Cortez, who's going to be talking about imaging mosquitoes.
So thank you all for being here, and I thank Remy for introducing today the mosquito. I'm also working with mosquito-borne disease, but in this case it's malaria. And I will talk about how I was involved in a project to really image the needle in the haystack, that is the parasite inside the mosquito. So, as I said, I work with malaria. There is still over 250 million cases every year of this disease, and it is transmitted by a mosquito of the Anopheles genus. It is also produced by a Plasmodium parasite. In particular, I work with Plasmodium falciparum, which is the most deadly human parasite. And as you see here, It is a fairly complex system. Don't worry, today we will only talk about these 3 stages. We will talk about the stages of transmission from human to the mosquito and then back to human. And they have something in common. What they have in common is secretory organelles. So these stages are called the gametocyte, the ocanid, and the sporozoite. And indeed, here in green you can see these secretory organelles are called different names, osmophilic bodies or micronins, but what they have in common is they are secreted to the outside of the parasite at moments of activation, and they are involved in processes of disrupting the cellular environment. So this gametocyte is inside the red blood cell, it activates, and then it needs to go outside the red blood cell. The secretory organelles have a role in this. The micronins of the ocanids are important for the parasite traversing the mosquito midgut. And the sporozoite is the same when it needs to traverse the human epithelium and then the hepatocyte to infect the human. Now, one more thing these secretory organelles have in common, and this is where my period came in, is they all have this protein called JES, which I am interested in. And then I wanted to study this protein, and what we do in the laboratory is let's take the gene out of the mosquito and then know what happens of the parasite and what happens when the parasite tries to infect the mosquito. And this is what we saw, that it was involved in all 3 stages. This is just comparison between wild type and knockout parasites. And for a good control, we reintroduced the gene just to know that the phenotype, the defect in these traversals or regress, that was due to the lack of this protein. So this protein really has an important role in transmission, but the question comes now, how it is involved in transmission. We want to understand the process here. For this, I turned to microscopy, and we have the luck to have a facility in our institute. And so starting with gametocytes, this is something that we can culture in the laboratory. We have flash, it grows inside red blood cells. You can culture and you can purify them to a decent purity. And this resulted in our local facility being able to image wild-type parasites. If you see there is nothing surrounding it, and especially there is no osmophilic bodies left, these secretory organelles. And this is the JES knockout parasite. Now the red arrow here, that signs the red blood cell. We expected that. We saw that already in our functional assays. But what about these osmophilic bodies? They are still here after activation. That really shouldn't happen. This could be something due to the lack of JES. But then I wanted to ask the same in ocanid and sporozoites. The problem is these guys, they only grow inside mosquito. We are not able to culture them. It's still not possible in Plasmodium falciparum. So that's what happened, and I did try to purify a little bit of this, and I gave samples to my local service, but it was just not possible to look for these parasites in electron microscopy. The samples were not good enough. And for this, I had a problem. My goal was to really understand what was happening to these organelles during activation of the parasite, and I needed to find the parasites within the sample. That's when ISIDORE came in. I learned about this fund scheme, and then I saw that there was a possibility to apply for Eurobioimaging for the hub. The people there was really helpful, really from the beginning, trying to make the project work, and they put me in contact with the BRAC node in Sexe Budečice with Marie and Jirka. They are the microscopists in Czech Republic that made this possible. Together with them, we built a project that they helped me completely to really apply for the correct technique that could work. And in the end, the goals were to localize the parasites in the sample, and we will use claim for that, and then to do 3D electron microscopy of these parasites. For this, we used 80 So, of course, what is all these weird letters? CLEM stands for correlated light electron microscopy. As I said, my parasites are there in the mosquito, and I need to find them to do decent electron microscopy of them. And as you see here, this consists of finding a parasite within the sample and then only imaging that little part. This is a mosquito midgut that it's in a grid, and then because parasites are stained and we can detect them, then we can really only acquire by electron microscopy the specific grid that contains the parasites. Once we detect where the parasite is that we want to acquire, it was done with 80 That's a radiomorphy scanning electron microscopy. In this case, several layers of the sample are acquired, and these are called ribbons. So they are sequential images of the parasites. In the end, we can reconstruct really a 3D model fully encompassing the properties of the cell. You can see here, let's see if this, yeah, this is a sporozoite parasite. You can see all the different layers. And then with some segmentation, manual aided by a bit of automation, you can really make a model which is the full cell in which we really can quantify these red dots. These are the microorganisms. So we can really analyze disposition, number, change of these in different samples. This is what we really wanted. The project was about getting images of all 3 stages, and we achieved that. Gametocyte, organ, sporozoites, samples are ready. And now the ball is back to me, and I'm starting to analyze this. So to show you that this really makes sense in an experimental way, we started with this. You may remember, this is really anecdotal evidence. It's quite difficult. I did try to quantify based on random sections of parasites. But now what we have, it's not only that, it's full models of parasite. This is before activation, and this is after activation of a wild type, with almost no eosinophilic bodies left, and then the knockout parasite with so many. That allows me to really put them into a comparison into graphics, and this confirmed really that our knockout parasites was not losing the eosinophilic bodies when activated. So this is so far where the project is. Conclusion is we have data from all 3 stages, and the analysis on gametocytes really proved that the analysis, the experimental approach is correct. What comes next is to quantify the organelles also in organids and sporozoites, and really to compare my wild-type parasites to the knockout. Importantly, this is also will serve as a reference data set for especially organids and sporozoites in falciparum. There is very sparse data on electron microscopy of these stages, and I don't think there is 3D electron microscopy on the public domain. And then on the more biological or more functional, what I want to do with the project now, I'm looking for protein partners of yeast. So to understand not only that, okay, yeast is very probably important for secretion of these organelles in the activation, but what it interacts with and what's doing at the molecular level. With this, I finish. I'm a postdoc at Max Planck Institute for Infection Biology. I would like to thank Elena, my PI, for hosting me and allowing me to do the project. Julia and Gemma were very involved in the project as well. And the people from Eurobiomi, special thanks to Arina, which was really making it super easy and super nice to apply and to get this project running. And then Marie and Jirka, which were the ones doing the microscopy. Thank you.
Thank you. And some of you may have been at the conference a year ago when we also heard about this, and so it's extremely gratifying to see the progress that's been made on that particular project. Time for some questions, if anybody has any. We need a microphone. So, Mabel,
Can you tell us some more information about the protein you're interested in? So, in terms of sequence, I don't know, domains, and what is your favorite hypothesis in its function regarding it?
So, the protein was studied in the murine model before, and it was detected only in gametocytes and sporozoites. It stands for gamete egress. That part was known already, but on the structure, there is no known domain. It has a transmembrane domain and a signal peptide, so we really assume it's going to the membrane, but there is no known other domain in the protein. This is a bit. We know that, for example, it's quite difficult to express them. We are trying to put them in vitro to see about some biochemical properties, but that the way might be to really identify the partners. For this, I'm doing some proximity-dependent labeling, trying to see what other proteins interact with it.
Okay. Thank you very much. So the last talk before the break, coffee break. Once again, we're now from a service provider. So Remy mentioned EVA, European Virus Archive, so the Misakova from EVA will be presenting online. So, are you there online? Yes.
Hi, can you hear me? Yes.
We hear you very well, thank you. We haven't got your slides yet.
Okay, let me see if we can fix that.
It may be at our end that needs to be fixed.
I see my slides. You?
Not quite. Yes, they've got a finger going up in the air, so it's coming. Yes. All good, thank you.
Okay. So we can start?
Yes, please.
Okay, thank you very much for inviting me. I'm here to talk a little bit more about Crimean Congo hemorrhagic fever, but this time in Corsica, in France. The presentation was kindly prepared by Paloma Coin. She's the third-year PhD student at the University of Corsica under the supervision of Alessandra Falci and Rémi Charel from the Marseille University. And I come from the provider's end, let's say. I come from University of Ljubljana, and we are partners of ISIDORE and EVA G, and we have a BSL 3+ facility in which we did the neutralization in this project. So if I start with a little bit of introduction, although it was already said today, the Crimean Congo hemorrhagic fever is emergent virus, and as you can see, due to its complex replication process, it's very prone to different errors, and this leads to significant genetic diversity. And up to now, we know for 7 distinct genotypes, and you can see that the genotypes are geographically, they have some geographical origin, but we can find different genotypes also in the different in the same, let's say, spots in the Europe and also in the Africa. So we don't find only European genotypes in the Europe, but we also find the African ones. So in the terms of geographic distribution, it's very diversely distributed. It's over 30 countries by now and over 15,000 cases. And this is probably due to its diverse tolerance to different environments, and also because intensified testing, as we heard already today, and also because of the broad distribution and spread of its primary vector. And its primary vector, of course, is the hyaloma tick. The hyaloma tick can reach to the new regions through various mechanisms. They may be transported by migratory birds or through the legal or illegal animal trade, and of course they are spreading. Of course, they are persisting in the new environments also because due to the changing environment and of course global warming. The natural reservoir is Hyaloma marginatum, and the ticks become infected at any life cycle stage during the feeding on viremic animals or during co-feeding with other infected ticks. And the humans are in a risk of infection mostly when they are encountered with some risk occupations, like farmers or workers working in the slaughterhouses. And the infection can be mild fever and with headache and myalgia, or very hemorrhagic form of disease with in some outbreaks with very high mortality. Like, for example, in Kosovo in 2001, the mortality was reaching almost 30%. So this is also, on one hand, probably because of some support treatment, and of course each outbreak has its own specific correlations, but also due to the different, might be also due to the different genetic lineages of the virus. So if we look at the project now, so in Corsica, is not just in Corsica, but all in other Western European countries, there is a growing concern of emergence of CCHF. As I said, we already found tick in various locations, but they also reported, especially after 2010, when they reported disease in Spain and Turkey. Corsica is a Mediterranean island. This means that it has a Mediterranean climate. It's nicely located along the migratory route of many birds, and it has a very well-known and established presence of Hyaloma ticks. And that's why they started with screening of cattle, sheep and goats, and they looked for serological detection of CCHF virus to see if they are like indicators of some virus circulation. And those first studies began in 2014, and by 2016 they had a quite significant prevalence in cattle and ruminants, but of course they didn't find any, in more than 8,000 ticks they tested, they didn't find any PCR positive ticks. So by the 2020, there was some evidence that CCHF or related virus might be present in Corsica, but no real PCR detection. So then in the study that I'm describing, that started in June 2002, they collected more than 5,000 ticks from a little bit less than 500 cattle that were in the slaughterhouses, and they found at two slaughterhouse sites, they found 119 positive PCR positive ticks. So they did sequencing on them, and they found out that the sequencing was actually the African genotype 1, which was quite identical or very closely related to the strains and to the genotypes that were found in Senegal in 1970s. And it was very distinct from the recent strains that were confirmed in Spain and now in mainland France. This study, this results presented the first CCHF virus detection in Corsica, and they also confirmed that what was previously suspected, that there is circulation of the virus in the region. So they moved on to the human surveillance study, in which they choose two populations, and one was a general population with 2,500 participants. They just collected the blood from the 11 different medical labs without any additional information, and the other population they tested was population that was classified as high-risk populations, so farmers, workers, woodworkers, and workers in the slaughterhouse, and workers in the hospitals. This was 201 participants, and they, besides collecting blood, they also collected data in a very detailed questionnaire and analyzed it questionnaire. And out of 201 participants, 65.9% reported recent tick bites. So a very exposed population. And they did ELISA, they did 2 commercial ELISAs in these samples, and found antibodies against IgG antibodies against CCHF in 2 individuals from the general population, in 2 individuals, older individuals that were from northern region. And in the at-risk population, they found antibodies in 1 53-year-old slaughterhouse worker who had previous tick bites and also experienced some fever after these tick bites. So when they got these results, they contacted through Remy Charel, ISIDORE, and through them University of Ljubljana, because University of Ljubljana has a BSL-3 infrastructure that provides safe and integrated solution for the work on the BSL-3 and also BSL-3, mostly BSL-3 pathogens, but we can also work with the CCHF, since the CCHF-40 strain was isolated in this laboratory and comes from Kosovo outbreak, which happened in 2001. And since then, the laboratory is operating in this region as a partner in clinical diagnostic of such cases. Otherwise, we are also working on isolating all other emerging viruses in the Balkan region, and we offer different services and also the cell lines through the EVA-G or ISIDORE projects. So we received from Corsica, we received the package with 38 sera that were either positive with Euroimmune or IDVAT ELISA, or they were 4 of them were borderline positive. We used for the VNT confirmation, we used the Vero S6 cell lines, and for the virus, we used our CCHF Kosovo-40 cell line, which is Europe 1. And the VNT, after the 7 days of incubation of mixture of antibodies and the virus on the cell cultures, we had 2 positive. One was a cattle with the titer 1 to 10, and one was this S1 sample, which belonged actually to this 53-year-old slaughterhouse worker that was positive actually against both, on both ELISAs, so Euroimmune and IDVAT. And his antibody was 1 to 40. And this what was already discussed today. We also found in our samples and the samples that we tested relatively low number of titers of neutralizing antibodies in CCHF infected patients, but we do see quite nice CPE, and we also, when we stain against N protein, we also see quite nice plaques. So the VNT really works nicely, and we are able to do quite, I think, sensitive investigation into neutralizing antibodies. So just to conclude, this study showed that although in Corsica there's virus present, definitely virus present in ticks, and there are antibodies, specific antibodies, both in animals and humans, they didn't find a acute human case yet. The possible explanation for that is that maybe they have to consider more testing in humans, especially in those that experience like summer fever, because maybe they have milder forms of CCHF. But also, if we look at other countries like Spain and Greece, we found virus in the ticks and some serological antibodies prior to the emergence of the first case. So I think that the important take-home message is that you have to be vigilant. They have to be vigilant in Corsica about the CCHF, and they have to continue. With the public health education, ongoing surveillance, and should be aware of possible future autochthonous cases in this region. And with this, with this, I would like to conclude my very brief presentation on this project. It was very nice collaboration, and I would like to thank all the people at the University of Corsica and also University of Marseille to approaching us, and I hope we will work on this subject more. So thank you, and I hope you have some questions.
Okay, so thank you very much indeed. It's now time for us to take a coffee break. So thank you, and we'll be back here at 4:10.
c'est pas toujours comme ça.
Et donc l'instruction, c'est si possible. Okay, we're going to be into the last session. So we're very pleased to have Antoine Gessin, medical virologist, studied in Paris hospitals, universities, MD, PhD, 2 years as a postdoc at the LTCB lab of Robert Gallo. Everybody knows Robert Gallo with his role in the discovery of HIV. And then a position at the Pasteur Institute, and he's been unit head since 2001. And so in addition to the work on human retroviruses, more recently there's been work on Fermi-retroviruses and monkeypox. And this is what we're going to be hearing about today.
Okay.
Thank you very much.
So thank you very much. Thanks for the invitation. I really appreciate to be here. I learned a lot of things. Thanks to Claudia also. And now I understand quite well what is ISIDORE. So I will, my talk will be quite different from the others. I will not speak about methodology, about the diagnostic test, about molecular biology. I will mostly speak about what is going on regarding monkeypox, everything you want to know without daring to ask, if I may say. And we will discuss about history, clinical, epidemiological aspect, the reservoir, and the current situation. So one word about the discovery of and isolation of monkeypox that occurred in 1958, and that was in a colony of an institute in Copenhagen, in Denmark, and there were 2 outbreaks of a spontaneous non-fatal pox disease in cynomolgus monkeys. That means monkeys from Asia. Quite important to understand, because, you know, monkeypox is strictly, occurs strictly in Africa. And there was a general maculopapular rash, and the general health of the animals were quite uninfected. There was no deaths, no fatal cases. And the isolation of the virus was done from pustular lesions in eggs, as you can see here. Here. And in tissue culture. At the time, there were no PCR and stuff like that, and it was not so easy to do sequence. And the guy demonstrated that these viruses were serologically related to vaccinavirus. You can see also they demonstrate that the viruses on electron microscopy has a typical brick shape. And this virus was caused monkeypox, despite the fact that now we know it does not really infect monkeys. Monkeys are not the reservoir of monkeypox, and it was only a pox virus, but nothing to do with smallpox. So just after that, for about 10 years, there was several other monkeypox outbreaks reported in captive colonies of monkeys in the USA, in the Netherlands, in France. There was no human cases detected during this outbreak, despite the fact that there was quite a lot of deaths in many animals affected. And this suggests it's quite important to understand that humans were not susceptible to monkeypox at that time, but it was very probably because all the personnel have been vaccinated against smallpox, like all the guy over 45 years here. The first case of monkeypox in human was after that reported in 1970 as part of the national smallpox surveillance, an eradication program then underway in Africa that was extremely important, the eradication, of course, of smallpox. This occurred in a small boy, 9-month-old boy, and he has a rash. You will see after that what kind of rash. And he was admitted to the hospital in DRC. DRC was Zaire at the time, belonging to Belgium, no more at that time, it was before. And he got what was quite important, painful cervical lymph nodes, which is quite different from varicella, and monkeypox was isolated from his skin lesion. Unfortunately, he recovered from monkeypox, but he died from measles, which is still quite frequent, as you know very well, in Africa for the moment in children. After that, what's happened? Human monkeypox, there were 6 cases reported mostly in children. You can see that they were in West Africa. Before that, we were speaking about DRC, Zaire, so in Nigeria, in Sierra Leone, and in Liberia. And what was important is that monkeypox remained an exclusively African disease with only sporadic cases in forested areas of Central and Western Africa, with small outbreaks in the human population, mainly in DRC, until 2003, when the first cases out of Africa were reported. What's happened at that time? Important to understand that at that time, the first monkeypox in humans out of Africa occurred in 2003, and this event occurred in the USA and was linked not to the travel of a guy infected with monkeypox, but of a transfer of the potential reservoir of the monkeypox. That was the importation of Gambian pouch rats, the Chrysotermis, from Ghana to Texas. These rodents transmit the virus to prairie dogs, you know, the small pets that you can buy in the USA. And they were housed in the same exotic animal facility, and after that, the virus infected humans, mostly young adults and children. There was no death, no hospitalization. That's important to understand because there was at that time about 80 cases, and when we are discussing now, there is hundreds of cases in Central African Republic and in DRC, and there is quite a lot of deaths. So at that time, few hospitalizations, no deaths, and monkeypox was isolated by cell culture, you know, from 7 patients and 1 prairie dog, and you can see here the primary inoculation reaction, usually by bites or scratch from the prairie dogs. After that now, the second out of Africa story was the importation out of Africa, the exportation out of Africa by human hosts. So quite different. Now we are speaking about humans who are living in Africa and that are infected by monkeypox. That occurs in 2018, especially with 3 cases in UK, 1 in Israel, 1 in Singapore. You can see here the different localization and the airport where they flew in Nigeria. And at that time, there was an outbreak in 2017, 2018, with about a little more than 100 confirmed or probable cases. It's not so easy to make the diagnosis at that time, even if it was only 6 years ago or 7 years ago. Important to understand here that these guys were not kids, about 30 years. And the distribution suggests both primary zoonosis, but it's always extremely difficult to demonstrate a link with an animal. And today there is no molecular link which has ever been demonstrated from an animal to a human regarding monkeypox. And for most of the other cases, it was probably human-to-human transmission. And most of them at that time were male, young, and most of them were also HIV infected. So you can imagine, despite the fact that, I'm sorry to say that, but you know, in most of the African countries, except probably South Africa, men who have sex with men is forbidden, if I may say, by law. So it's quite difficult to read in the papers if the papers were homosexual with a network, sexual network. Now, slowly by slowly, it appears that most of these persons were already homosexual with a lot of connections. So what's happened about 2 years ago when there was really a pandemic, an epidemic, a large epidemic of monkeypox. So a series of monkeypox cases were identified first in UK, after that in Portugal, in Italy, primarily in MSM. You can see there were some papers published. It's quite incredible because in about less than a week, the papers were published. And you can clearly see after that, so it was in May 2022, the health authorities rapidly established that we were at the beginning of the outbreak of a new clinical and epidemiological form of monkeypox, quite different to the situation that we knew before in Africa. So what is monkeypox? Monkeypox is belongs to the Poxviridae family, and to the genus Orthopox, like variola, vaccine, cowpox, camelpox, and recently the new Alaska pox. I have one slide at the end of the talk. It's a large envelope virus. Each variant contains about 200 kilobase pair, encoding approximately 200 orphan proteins. You can see the typical characteristic like ovoid bricks here. You can see on electron microscopy. It's very important to understand that orthopox are very similar genetically and antigenically, which explains the cross-reactivity regarding immunity with quite a lot of protection between them. To give you an idea, vaccination against smallpox generally protects against monkeypox, and in an historical study in Zaire a long time ago in Central Africa, there was about 85% of cross-reactivity and cross-protection. What do we know about the genetic variability of monkeypox? Historically, there are two genetic clade genomes which differ by less than 1%. The first being found in Central Africa, the second in West Africa. But with the emergence of monkeypox outside of Africa, and especially the need to destigmatize the disease and prevent discrimination, there was a lot of discussion about that. Definitions of two clades were done, clade 1 for Central Africa, clade 2 from West Africa. Interesting to understand that between Central and West Africa, you have few countries, and especially Cameroon. And Cameroon is the only place today where the two clades have been demonstrated. So probably there are different kinds of reservoirs in Cameroon. So let's speak a little bit about the reservoir of monkeypox. So monkeypox is a sylvatic zoonosis, but the reservoir is, for the moment, not yet clearly identified, despite quite a lot of papers regarding different possible reservoir or host, intermediary host. We know that probably rodents, various species of squirrel and rats living in the rainforest of Central and West Africa are among the best candidates. But just to give you an idea, African apes, monkeys, remember that monkeypox was isolated to the monkey at the beginning, but nobody knew exactly how the virus entered the colony, you know, in Copenhagen in 1958, probably from other animals in contact with cages originating from Africa. So African apes and monkeys are mostly intermediate hosts and not really reservoir, a little bit like, for example, like for Ebola, if I may say. Many animals, important to know also, like rabbits, prairie dogs, other rodents and monkeys are, however, susceptible to infection in captivity and in the laboratory. So what do we know? We work a little bit now regarding the possible animal reservoir. I think that just after me, there is a talk about the tropism in different animals. So we'll have something perhaps brand new just before me, after me. So we have done a study based on ecological niche comparison, and especially we compare the ecological niche of monkeypox virus to a very large amount of animals. We begin with 2,500, and after that we go to 250, and after that we have only about 100, exactly 99 mammals from Africa where we have sufficient points, you know, to draw a map of their distribution in Africa. And to make a long story short, so we published a small paper about that, but which is quite informative. And we believe that the best overlap was with this kind of small arborical squirrel, Phoenicirrus aneretris. We really believe that this is probably one or the reservoir for monkeypox. Now we are doing sequence analysis of the complete genome of a large diversity of such animals to do a phylogeography comparing the host and the viruses. What a little bit about clinical features. There are very few MDs here, but you will see what is really monkeypox and how it can occur. So it's important to understand that monkeypox in a classical African setting, this is completely different to the situation that occurred about 2 years ago with the epidemic in MSM. This is different, we'll speak that just after. Usually it occurs in small remote village in hard-to-reach rural forest area, very frequently in the context of armed conflict, like for example in some place in DRC, for the people who know a little bit that, population movement, usually with very poor medical infrastructure and also with poor patient care and follow-up. This explains very probably the much higher lethality than in other different places. That means also that the clinical and epidemiological data obtained usually, this has been done retrospectively, and in most of the cases they are incomplete. So you cannot really compare the situation about 2 years ago to the situation that occurred usually a long time ago and even now in Africa. Incubation about 13 days, 2 weeks, a little prodrome with a few days. After that you have eruptive phase, typical. It's a little bit like varicella, if I may say. And I'm not going to go through everything, but you have a centrifugal skin lesion, which is important, with different type of lesion, macule, papules, vesicule, and pustules. Usually they are frequently on the palm and soles of the feet, which is quite different from the monkeypox, or the chickenpox, because you can have both in the same population and even in the same individual. Usually also there are quite a lot of different lesions. The lesion usually at the same stage of development, you can have thousands of lesions, and the severity of the symptoms are proportional to the density of the skin lesions. After that, you can have crust, hypo or hyperpigmentation, as you can see here. Usually, in most of the cases, it is a self-limiting course, but you can have clinical sequelae, facial scars, and most of the severe complications are linked to bacterial sepsis. And in most of these small villages, quite remote, without nothing, or with nothing, if I may say, you can have a lethality not negligible, especially in children. And of course, if you have immunocompromised, especially HIV infected, it's usually quite worse, of course. So here, that was only, the different, you cannot see clearly. That was the most frequent clinical sign of monkeypox in the African setting. This is in Nigeria, West Africa. This is Central Africa, a study that we have done a few years ago and just published about last year. Important to understand that the case fatality rate of monkeypox in the classical African setting, usually we consider, if you consider WHO, for example, the case fatality rate appears to be lower in the West African clade than in the Central Africa. Nobody is really sure about that. Probably it's related to the situation regarding the healthcare system, generally speaking. It's about 2 to 10% or less in Nigeria, and more than 10% in DRC and Central African Republic. And as I told you before, if you are HIV and if you are a child, usually you have more. It's quite worse. So for the last 3 decades, there was a very large increase on the number of notified cases. It's important to understand that this is the notified cases, So probably before, nobody knows exactly what was going on in the field. DRC appears to be the most affected country. Just to give you an idea, 500 cases in 2011, more than 2,500 in 2018. And this year, from the beginning of the year to now, there is something like 7,000 cases reported in DRC, with more than 350 deaths, half in children. Important to understand on an epidemiological point of view, there is an increase in median age diagnostic over time. You remember that the first small child who get the virus at the beginning, about 50 years ago, was a very young one. After that, in 1970, 1980, most of the cases occurred in young children, and after that, it increased 20 years, 30 years, and now there's quite a lot of people with 35 or 40 years in Nigeria and in DRC. How can the virus is transmitted? The transmission could be from animal to person or person to person. As I told you, there could be a transmission, a zoonotic transmission via lesion or biological fluids from an infected animal, probably rodents, as I told you before, when hunting, butchering, or eating games. But as I told you, there is no virological confirmation of a single interspecies transmission to date on a molecular point of view. The human-to-human transmission occurs primarily through contact with biological fluids, especially the skin lesions. It's a little bit like varicella. And, but in most of the cases in Africa, the exact origin of the transmission is often unknown. So just to give you a few studies that we have done or others, you can demonstrate nosocomial outbreak, of course, in a small hospital or centre de santé, health center. Intrafamilial transmission, of course. The sexual transmission, for the moment, has been poorly documented, except a few months or a few years ago in Central Africa now. The transmission seen are generally short, maximum 5 to 7 events, and the classical reproduction numbers, the famous R0. Everybody knows what is R0 now, thanks to the COVID. Just to give you an idea, for measles, it's about 15. Huh? For COVID, it depends a little bit how you were living. But there, it is usually less than 1. That means there is no epidemic. Huh? Usually it's 0.5, 0.6, 0.7. Probably a little more in Central Africa than in West Africa. This has not been demonstrated. But as you will see just after, during the large pandemic that occurred 2 years ago, the R0 in some specific population with a lot of intersexual network was something like 2 or 3. It was the same disease, huh? The same virus, but a little bit clinico-epidemiological different. So what's happened in 2022 with the outbreak of monkeypox? The first cases of monkeypox in the outbreak was confirmed in the UK on 6 May 2022 in a man traveling from Nigeria. But importantly, unrelated cases with no documented history of travel to endemic countries like Nigeria were then reported in UK and Portugal, suggesting undetected local transmission, especially in these two countries, with earlier onset dates of symptoms in late April 2022, it's here. So we just discussed a little that before in another talk, but WHO declared a global health emergency on July 22. By September, which is about 4 months after, there was 80,000 cases reported, notified of monkeypox in human in more than 100 locations worldwide, with most of the countries having no history endemicity like USA, Spain, Brazil, or France regarding monkeypox. So it was really a pandemic. And the great majority occurred in men who identified themselves as men who had sex with men, reporting really highly hyperactive sexual network. that sustain STI. So the transmission here was through sexual contact. Just to give you an idea, in the first paper about the clinical story which was published in the New England Journal of Medicine, you can see it's a little bit difficult to see, but most of the patients were about 40 years. They were male, they were homosexual, and half of the patients, most of the patients were white at that time, and most of them, about half, I think, here only, were HIV. It switched a little bit. The epidemiology switched a little bit after this paper, but it remains relatively similar. So if you look at that time, in 2022, about the outbreak of monkeypox, you see here the distribution. You see here the daily confirmed case with really the huge increase, with a peak in about August 2022. The distribution in Europe at that time, and in France, that was the evolution, with the peak at that time. Most of the cases in France were diagnosed in Ile-de-France, in Paris hospital for most of the cases. So that's the only a little bit hard, if I may say, clinical story. So it was quite interesting to see that the clinical aspects correspond globally to the classical presentation, incubation, prodrome, and eruptive phases, but there was quite some difference that give rise to a new pattern on a clinical and epidemiological point of view. And especially that, as you can see here, there was quite a lot of lesion on the genital anals and perianal anals, areas like rectitis and oral lesion with pharyngitis. So that was a feature on the table with a comparison of the classical African and the new clinical epidemiological form of monkeypox that we wrote in the New England Journal of Medicine for the review at the end of 2022. So you can clearly see that that was quite different, as I told you before, regarding the location, the affected population, the epidemiological features, the transmission, the dissemination, the clinical phase, the symptoms, and even the two viruses were slightly different. We will speak a little bit after that. And the case fatality rate, about 1 to 15% in Africa, as I told you before, and very low during the large pandemic. So I like quite well this kind of story, you know, the natural history of monkeypox. So what was the situation? You have in the rainforest some Central and West Africa a reservoir that we don't know really for the moment. There was a small outbreak, as I told you before, depending on the rats from Ghana, where there have not been cases of monkeypox reported. That was about 20 years ago, and a little bit of epidemic with about 80 cases in the USA, no case fatality rate. You know, when you are hospitalized near the MIT or in the New England Journal of Medicine hospital, it's not exactly the same that in the middle of a remote area in Africa where you have nothing. And after that, from the reservoir, you can have zoonotic transmission. In most of the cases, it is in a child looking for food, and also with the hunters. And after that, you have a human-to-human transmission with a R0 quite low. So only small outbreak, few cases, and not at all hundreds of thousands of cases. It does not exist. So that was until 2016, 2017, and especially 2022, where there was an out of Africa exportation, probably, we don't know exactly, in few homosexual men who have been in Nigeria, and after that going to a large party, if I may say, in large Canary Island. And from this place, there was a huge dissemination all over the world. Just to give you an idea, 3 weeks after the first cases, there was already cases in Australia and nearly in Patagonia. Okay? So that demonstrates the rapidity of the network, and also that the world is a global village, a term that we have already used, you know, for the SARS one. So the phylogeny of the monkeypox virus strain, it changed a little bit, the viruses. As I told you before, there was one clade 1 and clade 2, Central Africa and West Africa. All the cases, without any exception of the pandemic, were the clade 2 from West Africa, Nigeria, but the clade now 2B, which is slightly different, especially because now, as the situation of there is only interspecies, there is no more interspecies transmission, there is only interhuman transmission, not from an animal to a human, but only within the human population, and there is action of epidemic 3, which demonstrate clearly, this is just to give you an idea, the clade 1 with very few G vari or C vari T, the clade 2A, the classical from West Africa, Nigeria at the beginning, but Ghana, Senegal, and so on, very little. And the new clade here, the clade 2B, beginning in 2016, based on the history of the most common ancestor, and now 90% of the sequence have the ApoB type 3 mutation. So what is the current situation in 2024? The epidemic is almost over in countries with high socioeconomic level. You can clearly see here, this is the daily confirmed cases, peaking in August, usually, and after that, now you have very few cases. It is not completely over. There are still some remote area, you know, for example, in Brazil, in US, in Europe, a little bit, but very few cases occur for the moment. And that's the same kind of things here. This is mainly due to information and preventive efforts, which is very important, and the use of therapeutics and vaccination in the population at risk, which were, as I told you before, gay, bisexual, MSM with multiple partners. It's interesting to see that the epidemic was remarkably controlled by the mobilization of targeted high-risk population, especially with behavioral modification to avoid monkeypox and the high uptake and effectiveness of the anti-smallpox vaccination. Just to remind you that there is different vaccine, but the most used one is the MVABM, which is the third generation of a live attenuated non-replicative modified Bavaria Ankara, which is made by Bavarian Nordic. And regarding the treatment, most of the people in our countries were treated, if possible, by tecovirimat, which was working quite well, especially to decrease the number of lesions and to decrease the transmissibility between humans. There was, unfortunately, some severe forms, especially during the pandemic, a little more than 90,000 cases, as I told you. There was a little more than 100 deaths, mostly in HIV-infected patients. So that was in Nigeria, a little bit different, but that was in different places in the world, Occidental world, with a higher mortality in people with more advanced HIV, especially if you have less than 200 cells by millimeters. That was a little bit obvious. What is now the current situation in DRC in 2023, 2024? Since December 2022, there was a new outbreak with an increasing number of suspected cases and deaths. Important to understand that it's not so easy to make the demonstration, the formal demonstration that you are infected by monkeypox in this kind of population. So like a little bit for Ebola, you have probable, suspected, and confirmed cases. So in 2023, last year, 15,000 cases, 600 deaths. In 2024, so this year, 6,500 suspected cases, 345 deaths, which is a lethality of about 5%. And most of the people are mainly children under 5 years old, and it occurs mainly in the Equator region of the DRC. You can see on this curve, that was 22, 23, I think. This is 24. And you can see the clear increase of, I cannot read, but I think there is a number of suspected cases and a number of deaths. So it's really something which is currently ongoing with a large epidemic locally. One word to finish about Alaska pox. I couldn't speak about monkeypox without telling one word about Alaska pox. It's I don't know. I don't think at all it will be something important, but at least it's important to know what's going on. In 2024, there was the first reported severe cases and deaths of the people in Alaska, in the region of Fairbanks. He was a patient with history of drug immunosuppression, secondary to cancer treatment, and he has a contact with a stray cat that regularly hunts small mammals and frequently scratches the patient. So, but before that, you have to know that there were 7 Alaska pox cases that have been reported in the resident of the Fairbanks area. For the people, it's not Anchorage, in the north of Alaska. And in these cases, it was usually, in these 7 cases, a self-limiting illness consisting of a localized rash and lymphadenopathy. And the CDC went on the topic, if I may say, and there was quite a lot of several mammals which were tested, and especially the redback vole. I didn't know what was that. This is the Campagnol à dos rouges. These animals are infected by this virus, but we don't know if it is really the reservoir. So in conclusion, I would say that the 2022 outbreak reminds us that viral emergence is a permanent phenomenon without boundaries, often unpredictable in its nature, target, and magnitude. It is very probably a never-ending story. So everybody here will have some work for years and years. Despite the drastic reduction of the 2022 monkeypox pandemic, don't forget Africa, because this viral sylvatic zoonosis, as now you know, will continue to increase and remain endemic there for a very long time, especially in high-risk zones like Central Africa. And the main risk factors are bushmeat hunting, malnutrition, HIV co-infection, and low level of primary health care. So how can you imagine that in a few months or in a few years you can modify something of these different risk factors? So what we need in area of endemicity? There is 2 things which are really lacking. First, the availability of a specific rapid diagnostic test, the typical point of care. This does not exist for the moment. And the second is the availability of therapeutic and preventive means, such as tecovirimat and other new drugs. There's quite a lot of new drugs ongoing. And the vaccination. You know, I just called to be sure one of my friends working at the INRB, Placide Mbala, who is the guy, you know, doing all the story in the field yesterday. And for the moment, despite the fact that we know that the tecovirimat is working quite well, it has been used with thousands of patients here, especially HIV guys. And for the moment, there is no availability of tecovirimat in Central Africa, even in DRC, where there is a big epidemic. And it's exactly the same. The MVA, the Bavarian Nordic, was available. It was free in France. It was not exactly the same, I think, in other surrounding countries for France. So most of the patients at risk went to France about 1 and 2 years ago to be vaccinated, and that works quite well, at least for a diminution of the spreading of the virus. So it is quite, so you know what you have to do here. A point of care box really is something which is quite important to develop. But it has to be nearly free and working not on the bedside, but in the middle of the jungle. So just to finish, I'd like to acknowledge the people who work with me on this project. You know, we begin to work, it was interesting for us because we begin to work about 10 years ago on monkeypox. So when the situation appears, pandemic, we were a little bit ready for many things, especially the Institut Pasteur de Bangui with Emmanuel Laconnet and his team. My team, so that's a little part of my team, but the people working on monkeypox with Nicolas Berthet, Manon and Olivier. The people working with Arnaud Fontanet for all the epidemiological stories, especially with Camille Besson, who has done the PhD, and Tamara also for all the anthropological studies, wonderful studies. Alexandre Assanin, who is a very good mammalogist and evolutionist at the Muséum d'Histoire Naturelle in Paris. And we are working, just to make a small joke, we're working quite a lot at the Tervuren Museum, okay, not far away, the African Museum, you know, because we are collecting, if I may say, a lot of specimens, especially from the squirrel, which are already dead for many, many, many years. So at least we are not going, you know, to Central Africa to collect quite a lot of sample. And also Jean-Claude Maniguera and Yazdan Pana, the boss of the NRSME, we wrote with me the New England Journal of Medicine review. Thank you very much.
Thank you very much for that very broad overview. Questions? Barry.
Thank you very much. I was wondering, in the outbreak outside of Africa, it was clearly affiliated or associated with a change in epidemiology, change in transmission. Yes. So what is the cause of this huge outbreak now in the DRC? Is there also a change, or is the virus different?
That's a very good question. It's not really a change in the virus, despite the fact that now, because there is a little more human-to-human transmission, you can see the Mpox story and this kind of thing. So there is small variants occurring. But this is probably due to two things. But the main thing is the better knowledge about monkeypox for the last few years, the availability of a little funding. More people are working on that. You can now at INRB do direct sequencing with a MinION, for example. This is working quite well. Even it was the same, you know, for very remote area for Ebola about 2-3 years ago, when in a few days they can do these kind of things. So it's a better notification, more people working on that, better funding. This is very probably the main situation. And slowly now, you know, there is not a single person which is still living, if I may say, with protection, or even you need to be at least more than 60 years old or something like that.
Well, thank you. So in your very beautiful image depicting the natural history of Mpox, there you made a point that zoonotic transmission may result from hunting activities. But then I would imagine that in the villages you would also have rodents, other rat species. They don't play a potential role as reservoir species? Or?
Okay, there is something which is a little bit different to understand with monkeypox. You know, for example, I work a lot on HTLV1 and other retroviruses, which is the last chronic infection. So even 20 years after, you can have a trace of being positive, because you can be positive for the virus and positive for antibodies. There is no specific antibody serological test for monkeypox. Okay? That's it. There is a kind of cross-reactivity with all orthopox. So you can be infected by camelpox, by many, many, many different, and specifically all the viruses that you don't know yet. So it's difficult to make epidemiological studies regarding these kind of things. And we believe, but probably we are wrong, that it is an arborical squirrel. So most of the people in Africa working with rodents, squirrel are rodents, trap animals in places where the people are living, okay? Because they work on different viruses like antivirus or you know, Lassa virus and things like that. So it's not so easy to have this kind of samples first, because it's in the middle of the trees, and this is one of the answers. Okay. Yeah.
Okay.
Okay.
Many thanks.
Thank you very much.
So I hope Paul Groma is online. We'll be staying with monkeypox.
Yes, hello, I'm online. Can you hear me?
We can hear you very well.
Okay, great. So welcome and thank you for giving me the opportunity. to present our ISIDORE project. My name is Paul Groma. I'm a senior social scientist at the Medical University of Vienna. And I'm going to report about a reflection process on the Mpox response that happened in 3 European countries. So my presentation is also about monkeypox, but I will call it Mpox in my presentation. And I guess my presentation is a little bit different to the presentations we had before, because we are not having a medical approach to this subject. We are social scientists, so we're not looking at the virus itself or at the vaccine, but we were looking at society, how society reacted to the outbreak, and how certain communities and community actors were engaged in tackling the disease by providing information on the virus and on the protection, and how they were directing patients to care. So, how can I? Okay, I'm here. Thank you. So a bit about the background, who we are. We are from the Medical University of Vienna, from the Center of Public Health. And we are medical anthropologists working at the Unit for Global Health. The study was done by Ruud Kutelek and myself. And the TNA project was initiated by WHO Europe, the RCCE unit, or the risk communication unit from WHO Europe. And our collaboration started with a GORN collaboration, that's the Global Outbreak Alert Response Network, where we assisted WHO in developing a community engagement strategy on Mpox. And this was at the very beginning of the outbreak it started in June 2022. And the collaboration lasted until October. And after this collaboration ended, we suggested WHO to start this transnational access, or TNA project, with ISIDORE. So we were paving the way for WHO to seek this collaboration. And the TNA itself, or this reflection process, was realized in 2023 and 24. And the preliminary results were presented early this year, but the final results are still in preparation and will be published probably by the end of the year. Next slide, please. Can I navigate myself, or is it someone else navigating? Okay, so about the project. The project is called Reflection Process on the Community Mpox Responses in 3 European countries. And this happened because WHO Europe wanted to review its newly developed RCCE strategy, which means risk communication and community engagement, including infodemic management. And this strategy was first put in practice during the Mpox outbreak in 2022. And to see how it worked, WHO Europe was looking for an independent scientific partner to assess their approach with a qualitative social science method. They wanted to avoid the feeling of control or evaluation. So that's why we tried to find another approach, which we called reflection process, where we collected stakeholders' opinions and experiences with the Mpox response. And we involved practitioners from 3 European countries in a reflective feedback dialogue. And practitioners, they were, for example, public health authorities, specialists for sexually transmitted disease, community doctors, which work closely with the LGBT community, but also civil society organizations who deal with sexual education and LGBTIQ issues. Next slide, please. Thank you. What were the objectives of this reflection process? We wanted to see how the stakeholders experienced access and dissemination of information materials. These Mpox information materials were developed by ECDC and WHO Europe in a collaboration, and they included guidelines, for example, for event organizers for LGBT events like the how you call it, rainbow parades, gay parades. There were big happenings in many European cities at that time that also contributed to the spread of Mpox. And there we wanted to put our focus to prevent the virus from spreading. So we produced these guidelines, a number of leaflets, a number of social media materials to be distributed at the respective communities. Then we wanted to know how understandable and practical these materials were, and if these materials led to the implementations of local community campaigns, and if not, what were the reasons why people found these limitations. Then also we looked at the management of misinformation in the 3 different countries, which really was a big issue with regards to media reports on Mpox and discriminatory language used in the reporting and also in the basic information on Mpox. Also in this process, in this reflection process, we collected feedback and recommendations, how the materials, but also the approach to reach the respective communities could be improved. And as I said, these materials included materials from ECDC as well as from WHO. Next slide, please. Well, the results were presented in a comprehensive report, and they were circulated internally with WHO Europe. And they also, WHO Europe also called for informal meetings with their CSO partners. And this is still ongoing, and the results are being shared in this process. Unfortunately, we could not publish our comprehensive report, but we're now working in a scientific publication. But I will come to that later. There are certain restrictions that come from the collaboration with WHO, why we cannot present or publicly present everything that we found out. And so we have to issue a report that is very diplomatic and does not reveal which countries were investigated or which stakeholders took place. So that's why I cannot present the results here, but I'm ready to respond to any questions in this conference. But now my presentation will more focus on the process of collaboration between ISIDORE, the medical university, and WHO. Next slide, please. Well, for this slide, I must say I'm only the messenger, because I was only involved in one meeting between ISIDORE and the Medical University of Vienna. It was mainly our project manager that continuously had meetings with ISIDORE throughout the whole project. But we had several meetings to clarify our eligibility and the terms of conditions for TNA, how it would be possible to invite WHO or Europe, or to tell them that this collaboration possibility exists. Then there were further meetings between the TNA partner, or WHO Europe, and the work package lead, or the ISIDORE management, so that the application procedures for the TNA partner were settled. And also here, I'm just a messenger because I was not concerned with it, but we were also facing challenges because the administrative efforts that we were facing in the TNA were quite different to the administrative requirements of the Medical University of Vienna. So they came up with a lot of questions to us, and we had a lot of additional admin requirements. And this was a bit difficult to handle for our project manager. So the next slide, please.
If there are going to be, sorry, just to let you know that if you want to have time for questions, you'll need to finish in about 1 minute.
Okay, so then I keep it short. Maybe then we skip this slide and we go to the next one. I just want to go into the collaboration challenges that we had with the WHO. As I mentioned, WHO has a principle of impartiality and confidentiality, as we also have as a medical university. But it required a degree of anonymization and that we did not expect. So this was really a challenge for us. And also for the protection of participants, it was far more elaborate than we would have anticipated. Yeah, also there was a potential bias in the selection of participants because WHO proposed as participants they had collaborations with, and they already provided them with information materials, and it would not have given us a complete picture if we only interviewed these persons. So we had to select additional informants not to have this bias. And also we experienced that a lot of informants denied to take part in our study because potentially they feared consequences of reporting on their collaboration or non-collaboration with WHO. So the last slide, and then I'm through. Yeah, what is the sustainability of this TNA collaboration? What will be the next steps? So we found out that WHO Europe is quite happy with our results, and they are going to integrate our recommendations in their risk communication policy for the future. So they want to establish a permanent CSO representation in public health structures so that the access of CSOs to deciding mechanisms will be strengthened. And what else? Yeah, we have now established a basis for cooperation with WHO Europe. So this TNA will probably have follow-up projects. And yeah, and of course, very wealthy was that in all 3 countries, we met a lot of civil societies and organizations and experts from the sample countries, which also gives the possibility of future research projects. Then we have our publication in the pipeline. And yeah, I hope that my presentation will also inspire another some similar projects to collect experiences with community engagement approaches. So I want to stop here and have more time for questions. And I'm ready to answer them as for now. Thank you.
Thank you very much. Despite skipping over a slide, we are very short on time. So if there is one burning question, we could take that. Otherwise, I think from what you were saying on the administrative side, so just on a very practical level, we'd be very interested in learning what lessons you learned about putting the TNA procedure in place within the university, and whether that's information that could be shared with other administrators to help that procedure. But that's something we'll probably follow up with you directly.
Yeah, that's what I tried to line out in my presentation, because our details are too detailed and too specific to put it in 10 minutes. But really, for us, it was a very interesting collaboration. And for our own university, it was, of course, a benefit to have this contact now with WHO Europe. And despite there were some limitations, it was a very fruitful collaboration. And we hope we can continue with similar projects.
Excellent. Thank you very much. So we're going to stay with Mpox, more on the experimental side now. Barry Rox can tell us about virus host range.
Thank you very much. I'll wait for the slides, but I'd like to thank the organizers for the opportunity to present some of our data. My name is Barry Rox. I'm at the Wageningen Bioveterinary Research Institute in the Netherlands, part of Wageningen University. And we were involved in this project as service providers. And I'm presenting on behalf of Laurens Liesenborg and his team at the Institute of Tropical Medicine at Belgium, who unfortunately could not be here today. And the project that was funded was determining the Mpox virus host range in response to the outbreak in 2022. And I'd like to thank the keynote speaker for a very nice introduction, so I can really skip through all of the intro of Mpox and go straight to what we're planning to do, which is looking at the reservoir. And as the keynote already addressed, in endemic areas, small rodents are believed to be reservoirs. And as I just heard, it's probably most likely the squirrel that is the main target species. But really in Europe, it's not known. Previously, no one really bothered because it was not a problem. But in 2022, with the widespread transmission of Mpox in Europe and beyond, there was a concern that maybe Mpox could also establish in Europe. And which species that could be was not really known. So we already saw some examples in the keynote on potential transmission cycles, for instance in the US from prairie dogs to humans, but also with other pox viruses. There have been examples of transmission from animals to humans, including from livestock, but also not just directly from livestock direct contact, but also through potential rodents, either wildlife or maybe pests that are in the presence of these species. And so this led us to think about what kind of species could potentially play a role in Europe. And there were a couple of examples that I just wanted to emphasize. There's been evidence of cowpox virus transmission from rats to monkeys in the Netherlands. And this was a monkey colony that became infected with Mpox, and when they started surveying rats that were in the neighborhood, they found that that's where the virus likely came from. There's also evidence of rat to human transmission of cowpox in the past. And in the current outbreak in 2022, there's been a lot of evidence of or detection of monkeypox DNA in wastewater samples, suggesting that this virus is at least shed and potentially could play a role in transmission through sewage. And this was particularly interesting because one of the risks that we were thinking about was that maybe rats within the sewage system could take up this virus and then subsequently spread it to other species. And so the research question for this, the call that came out on Mpox through ISIDORE was to see which animal species would be susceptible to infection with Mpox and whether they could play a role in the transmission cycle as a possible reservoir. And we had ultimately, after a lot of discussions, we ended up with 3 different species. First of all, the rats. As I mentioned, rats are in contact with sewage. There's virus present in the sewage, so that could be a route. And as was known already from literature, rats are known to transmit other pox viruses to humans and other species. So that was a main focus, and that will also be the focus of my talk today. But part of the project was also we would be interested in studying whether cats could play a role, because cats could actually play a role as a bridging species between rodents, catching rodents, and humans or livestock. And cats are also known to be susceptible to, for instance, cowpox virus. And then ultimately also cattle, due to contact with humans, but also the presence of potential rats or cats on the farms. And of course, we know that cowpox is also susceptible, or that cattle is also susceptible to cowpox. So for the purpose of today, I'll talk about the first experiment that we performed through this TNA, which is infection in rats. And actually, there's been a study in 1976 where they showed, or where they tried to see whether Mpox could infect different animal species, including rats. And in this study, they used adult white rats and newborn rats. And when they inoculated adult rats, either through intravenous, intranasal, or scarification, they did not see any clinical signs, and they could not detect any virus in the blood, any tissues that were sampled, or in the skin. But there's one caveat, or actually two caveats. One is the sampling. They only took weekly sampling, so it could still be that if it's an acute infection, that we would miss it. And the other thing is, in 1976, the methods of detection were not as sensitive as we have today. The reason why we still wanted to stick with rats is actually because when they did it in newborns, intranasal inoculation resulted in a lethal infection within 5 to 6 days. So this suggests, and actually in that experiment they also showed that they could find infectious virus in different tissues. So this at least suggests that the rat has at least the possibility of becoming infected. So our hypothesis was that maybe in this experiment the sampling was just too broad and we would miss acute infection. So that's what we set out to do. And so again, the objective here specifically was to study the reservoir potential of rats to Mpox infection. And what we did was we took female rats aged 4 to 6 weeks at arrival, and we put them in two groups, either a group challenged with the Mpox virus or a control group. And we euthanized groups of animals on days 2, 10, and 21. Day 2 was selected as an early time point to see if there's really an acute infection, whether we could still detect it. Day 10 we figured would be sort of at the end of the infection. And day 21 group was chosen to follow up animals for 3 weeks to see if there's any clinical signs and so forth. We used a clinical isolate, clade 2b, so this is the virus that was circulating in 2022, and this was isolated from a patient in Belgium, and that we got this from the Institute of Tropical Medicine in Antwerp. We did 2 routes of inoculation in the same animal, so we did an intranasal inoculation by depositing virus inside into the nostrils, and then on the same animal we also inoculated via scarification using this bifurcated needle, which is being used for vaccination against smallpox. We did that on a site on the back of the rat. And then go straight into the results. So we looked at weight loss as a measure of clinical signs, and we saw that the Mpox inoculated animals did not show any weight loss compared to our control animals. We also looked at temperature to see if there would be a fever as a measure of or evidence of infection, but we also did not see any increase in body temperature over time between Mpox or control animals. But what we did see was, starting at day 4, we started to see lesions at the site of inoculation on the skin. And I hope you can see that here, but here you can see some lesions. Now, I should say, these are not, they did not follow the standard development of lesions that were discussed earlier with the different stages. That is most likely, I think, because of the way that we sampled these animals, because we actually, every other day, we took quite robust skin samples by swabbing the skin. So this probably resulted in some damage of the lesions. So that's probably the reason why we didn't see the different stages that have been reported. These lesions were detected up to day 11 post inoculation, and were only present at the site of inoculation, so they did not spread. And by day 14, the lesions were mostly gone. There was no gross pathology that we saw upon necropsy on day 2 or day 10, other than the skin lesions. So again, here, this is on day 10. You still see some remnants, some scars of the lesions that we saw in the skin. Then we also looked at virus shedding, because of course, if the rat would play a role as a reservoir, it should also be shedding virus. So interestingly, here, this is in throat swabs. We could see by day 6, we see high levels of virus, but it's very transient. So by day 10, it's already gone. I think what's interesting is on day 2 and day 4, these samples were negative. So this really suggests that there's replication of the virus, and it's not just the inoculum that we're picking up. The same with nose swabs, although it's much lower compared to the throat swabs. In one animal, we found, on one day, we found a virus in a rectal swab. And then also here on the skin swab, this is quite variable, but in all animals, we could find at some time point a virus or viral genome in these swabs. And then I should also emphasize in the throat swabs on day 6 and on the skin swabs on day 4, we were also able to isolate infectious virus from these samples. Then we also looked at replication in different tissues. For the nasal turbinates, all samples were positive on day 2 and day 10. And we can see here, day 2 was slightly higher compared to day 10. And all of these were also positive by virus isolation. In one animal on day 2, we found that there was virus present in the lungs with quite a high CT value of 37, so not much virus present. But any other samples, so no evidence of viremia over time. And then on day 2 or 10, there was no virus detected in brain, skin, kidney, liver, spleen, etc. So the skin was actually particularly surprising since we did find the lesions. Then we also looked at pathology. So the skin lesions that we saw on day 10, we saw evidence of inflammation and some necrosis of the keratinocytes. The only other thing that we saw was that there were enlarged cervical lymph nodes, which are the draining lymph nodes for the skin. Yes, almost done. And no other gross pathology was found. And again, quite surprising, we didn't find any evidence of viral antigen in any of the tissues, so neither in the lung, the nose, or in the skin. So in conclusion, we showed that rats indeed are susceptible to Mpox infection, but the infection is quite acute and self-limiting. They develop skin lesions, but no other clinical signs, and that infectious virus is shed by the respiratory tract and by skin lesions. The susceptibility of Mpox and the presence of infectious virus suggests that the rat could potentially play a role in transmission, but the efficiency of transmission is not really known, and that's also not part of the study design, so we don't know if there's animal-to-animal transmission. But based on the acute self-limiting infection and the lack of viremia and the lack of prolonged shedding, we think that there's a very limited risk for the rats to play a role as a reservoir for Mpox. As part of this project, we still have plans to also assess the susceptibility of cats and cattle. We'll start at least the cattle experiment is planned for in September. So hopefully, perhaps the next time we meet, I can present the rest of this data. And with that, I'd like to thank all the people. So my team at WBVR, the team from Laurens at the Tropical Medicine Institute, and then also people from RIVM, the Dutch National Institute for Public Health, who were also involved in the risk assessment. And of course, the funding through ISIDORE. With that, I'll be happy to take any questions.
Bata?
Yep, I have a small question. So monkeypox is not known as a respiratory virus, and yet you use the inhalation route. And in another slide, you showed that monkeypox infection could be acquired through eating contaminated products. So I'm just wondering why you didn't use the oral route instead of the nasal route for inoculation of your rats.
Yeah, so there actually is evidence that it can also be respiratory, but at least for the most part, this is shown through experimental settings. So one of the people that was involved in the early steps actually did some intranasal challenge or respiratory challenge of Mpox in non-human primates and showed that this was very quite efficient way to show. And I think also in humans, it is possible, but it's not the most the most used route. Yeah.
And I know we had discussion already, but do you think squirrels would have been a better choice to try to know if there could be the reservoir in Northern Europe?
Yeah, we thought about squirrels. The thing with squirrels, at least in Europe, is that there's not a lot of contact between squirrels and humans. So then it could still act as a sort of intermediate, but there was the main reason not to choose, other than the practicality of not having access to squirrels in our facilities.
So it seems that you have a productive replication, but self-limiting. So I was wondering, have you looked into the innate immune response, or are you planning to do that to see perhaps if just the innate antiviral response could limit the replication?
We haven't looked at it yet. No, that was also not part of the application. I think it would be interesting. I'm just wondering whether the samples that we have are the best for this, because in the end, we beforehand, of course, we have to decide on time points, and in hindsight, I probably would have changed some of the timing, because it seems that we were right before and right after the actual replication in different tissues. So I do think there is replication in some of these tissues. We just missed it in our time frame.
Very quick, please.
Yeah, just a quick question about gender. So as far as I understood, men are more likely to get Mpox. What about the reservoir? Do you test it for if there was any difference between like male and female?
So two answers. One is I don't think men are more susceptible. I think it's more the way that the transmission works, but perhaps you know better. Okay. And the other question, or the other answer, is mostly practical. It's a lot easier to co-house females than it is to co-house males in our facility. So it's more of a practical that we can actually house animals in groups rather than individually.
Okay. I think that's fine.
Thank you.
Thank you very much. So now we're on to anti-TB drugs. Nibitieta Rath.
Good evening, everyone. So today I'll be talking about, next slide. So I can do that, right?
Yes.
Okay. Randomized trial of metformin with the anti-TB drugs for early sputum conversion in adult with PTB. So before getting into the topic, I'm from Open Source Pharma Foundation, so which is a global non-profit. We're based in India, Paris, and New York. And as you can see, our motto is medicine together and medicine for all. This is some of the press clip about OSPF and some of the award that we have received. And this is our blue chip funders, Rockefeller Foundation, Tata Trust, Welcome, and national government agency as far as collaboration is concerned, and legendary scientists on board. So this trial, what I'm going to talk about, this is the three publication that I'm showing, and the another one is in the pipeline. So the first and foremost question, so why metformin for TB? So we all know innate immunity plays a key role, and immunomodulator or the host-directed therapy, they hold promise. So metformin is a very small molecule. It's a biguanide, and it is a very widely used type 2 diabetic medicine. So it has significant preclinical evidence that it works for TB. So it's an immunomodulator and enhances intracellular killing of TB via ROS signaling pathways, AMPK pathways, and autophagy. And also it enhances in vivo efficacy of first-line regimen of TB drugs. And it reduces TB-induced lung pathology. So this slide, as you can see, metformin as an adjunct therapy in MAC mice in TB patients. So when we treated, this got published by Singhal in 2014. And in the macrophage, what happened when you treated the different strain of MDR, with isoniazid and different concentration of metformin, what we observe is by increasing the concentration, you are also decreasing the survival rate of MTB strains. So when the mouse model were treated with isoniazid and metformin, 42 days after the infection, what we have seen that metformin and isoniazid combination is reducing the lung bacillary load in comparison to the isoniazid alone. So isoniazid is one of the first-line regimen for TB. In the retrospective study, in the retrospective clinical trial, you can see here when TB patients with type 2 diabetes were treated with metformin along with the first-line regimen, and in the another arm, in the control arm, with other diabetic medicines and the first-line regimen, we have seen that the lung cavity closure was faster in the metformin arm in comparison to the control arm. So all these things encourage us to go ahead with the phase 2B clinical trial. And how we can get into phase 2B clinical trial without even doing a phase 1, because this first line regimen are already there, which is being used for last 40 to 50 years. And we are now adding another medication, which is metformin, which is already being used for last 60 years for type 2 diabetics. So the primary objective was to evaluate the antibacterial activity of metformin when given along with 8 weeks, along with the standard TB treatment. So let me tell you the first line regimen for TB. It's isoniazid, rifampicin, ethambutol, and pyrazinamide. And the secondary objective was to evaluate the time to detection in culture, to determine the percentage of patient with the sputum culture conversion, to study the pharmacokinetics of metformin along with the first-line regimen, and to assess the safety and tolerability of metformin, and to study the immune response in the study subjects. So this was a multicentric trial which was done in India. So NIRT was our primary collaborator, and we had site All India Institute of Medical Science, maybe some of you already know, then site at Vellore, Madurai, and Pune. So as you can see, it was a randomized open-label parallel arm controlled clinical trial. So the first 2 months were HREG plus metformin, and followed by HRE for the next 4 months. So TB treatment is usually, for drug-sensitive TB, it is 6 months treatment. And then the secondary, and for the control arm, it was HREG for first 2 months and HRE for the next 4 months. I'll skip this one. So the current start of the trial, we have already completed the trial. We enrolled 324 TB patients for the trial, 6 months follow-up after the treatment has been completed. Lab result like sputum bacteriology, PK, pharmacogenomics, autophagy, everything has been analyzed, and also the PFT study has been done. But we are under analyzing the PFT study, and it has been submitted now for publication. That's why you don't see that paper there. So this is the study diagram. So what we did is, as you know, metformin is a type 2 diabetic medicine, right? So we started with 500 mg the first week, and then we shift to the 7 weeks of 100 mg, 1,000 mg. And then here, what I'm trying to show you, like from we selected 550 new sputum smear positive TB patients, out of which 160 were got enrolled for the metformin arm, and 162 were in the control arm. However, 155 in the metrif arm and 151 in the control arm were included in the final analysis. So this is the median time to sputum culture conversion by MIGIT. It was 42 days in the metrif arm and 41 days in the control arm. However, in the LG medium, what we observe is it is 35 days in the metrif arm and 29 days in the control arm. So there are a lot of factors that are associated with faster sputum culture conversion, and one among them was the higher body mass index. So when there is a higher body mass index, it is associated with the faster sputum culture conversion, and whereas the alcohol consumption was associated with the longer sputum culture conversion. This is the adverse event. Definitely, we have observed some SAE and AE during the trial. So out of the 10 serious adverse event, 8 were in the metrif arm and 3 were in the control arm. So arthralgia, which is the joint pain, it was mostly in the control arm, whereas pyrexia was in the metrif arm. And in the metrif arm, we have seen like gastrointestinal discomfort. That was one of the side effects of metformin. So here we can see that acute phase proteins and inflammatory cytokines. So this is one of the findings that encourages us to go ahead with the phase 3 trial and along with one of our chest X-ray. So if you can see here at the baseline, CRP, A2M, then SAP, and HP were normal for both the arms. However, after 2 months of the treatment, in the METRIP arm, they have been decreased significantly, whereas in the control arm, in comparison to the control arm. Similarly, the inflammatory, pro-inflammatory cytokines like IFN gamma, TNF alpha, IL-17A, and so on, they were same at the baseline, but after 2 months of the treatment, they were diminished significantly in the METRIP arm in comparison to the control arm. So pharmacokinetic parameters, so what we found is the Cmax of rifampicin and isoniazid were not significantly different between both the arms. However, the clearance rate was higher in the METRIP arm in comparison to the control arm. So the radiological improvement, what we have seen is in the METRIP arm, the radiological improvement in comparison to the control arm, it was statistically significant. So this gave us hope and the courage to go ahead with the phase 3 clinical trial, which I'll be talking about. So the summary and implication is metformin addition was associated with the faster resolution to the cavity on the chest X-ray and reduction in the level of the circulating plasma and pro-inflammatory cytokines. And after 8 weeks of treatment. So remember that here we are having the metformin intervention for first 8 weeks. And the treatment duration for the TB patient is for 6 months. And this suggests that a metformin-containing fried drug regimen may reduce the infectiousness and the transmissibility in patients with the cavitary disease, and it may also play a role in reducing the post-TB long sequels, mostly the fibrosis, long fibrosis, and structural damage. So definitely we didn't achieve our primary endpoint, which was it didn't affect the 2-month culture conversion, so definitely didn't address the question of shortening the duration of the treatment. So this is where we reach out to ISIDORE. So what we did is we, thanks to Patricia and David for putting us in touch with ISIDORE, and we wanted the regulatory advice very badly, just to figure out what next, what we should do. And this is where Dr. Byrne, he gave us this advice. He asked us to come up with a target product profile for metformin, and also that he suggested that it should contain, the TPP in defining the development strategy, study protocol design, and so on. And the key component of a TPP should include the product detail, dosage, regimen, target indication, and so on. And also he advised us to design an adaptive trial with blinded power analysis. Also, he suggested to have a site, he emphasized on having a site in Africa. And I will now show you what we have done after that advice. So it all happened in a very short period of time, maybe within 6 months, we reached to ISIDORE and then got into all this step. So we have already made the TPP for the first draft, I should say, even though it needs a lot of work, the first draft of TPP for not only metformin, because another medicine we are trying to take for phase 3 trial, which is N-acetylcysteine. And also we are making TPP for host-directed therapies looking into the lung function. So this is what we have come up with now. We have formed a consortium. We realized that without a consortium, single-handedly, we can't do a global trial. And the reason for doing a global trial not being specific to India, because if you want to roll out, if the trial being successful and if you want to roll out, you have to have a global trial. So we have formed a consortium with TB Alliance and ICMR NIRT, which is our collaborator in India for all the trial that is happening in India. The Aurum Institute, which is based in South Africa, Agency for Science and Technology and Research, which is in Singapore, and Newfound. So today I'll be talking a little bit about the Newfound. So the Newfound is an organization that we have formed. It's a collaboration of NIH, the ITRIS, and Fiocruz Brazil and OSPF. So this is the organization which is going to focus on repurposing for rare diseases and other neglected diseases in very short. And you will see more if you go to the website newfound.med. So this is what we are proposing after the this is what we are proposing after the ISIDORE advice. We are proposing to have a randomized clinical trial to evaluate the effect of N-acetylcysteine, which is also a generic drug, and metformin in reducing the lung function impairment among newly diagnosed adult PTB patients. And the rationale for using NAC and metformin I have already mentioned, and NAC, as you already know, it has been used for such a long, several decades. It's a muculating agent, and also it has shown different efficacy for lung function. So this is the antibacterial mycobacterial mechanism through immunomodulation, enhanced GHS label, and also a direct antimicrobial activity. So our primary objective, just keep in mind that this is not the final, because we are going ahead again to Dr. Byrne for the advice to figure out whether this looks okay, or if not, then what modification we need to do. So the primary objective as of now is to evaluate the effect of NAC and metformin in reducing the lung function impairment at the end of 1 year post-successful treatment when given along with the first-line anti-TB drug for the newly diagnosed sputum-positive PTB patient. The second objective is to compare the following two sustained treatment success after 1 post-year treatment and median time to sputum culture conversion, and the extent of radiological clearance at 2 months and at the end of the treatment, and proportion of the patient with the adverse drug reaction, immune response, and drug pharmacokinetics, and interaction between metformin, NAC, and ATT. So this is what we are proposing as far as the study design is concerned. NAC, 2 months of NAC along with the 2 months, the first is 2 months of NAC with HREG, and then followed by 4 months of NAC with HRE. And similarly, 2 months of metformin with HREG, followed by 4 months of metformin with HRE. And in the control arm, 2 months of HREG and followed by 4 months of HRE. This is, we just drafted this based on the India protocol, but as I speak, yesterday we had a call with the Brazil team, and the Brazil on board now, because we want a site in Brazil. So Brazil is on board now for this trial, and also we are looking for site in Eastern Europe and also in South Africa and Africa. So the study duration would be 3 years, recruitment 1 year, 6 months post-treatment, and 12 months post-RX follow-up. So this is where I'd like to stop, and if you have any question. Thank you, everyone.
So I'm afraid in the interests of time, we're going to have to move on.
Okay.
And so thank you very much. And we're moving on to the last presentation. Mia Lopez Siles, who's going to be talking about Klebsiella.
Good afternoon, everyone. I will be presenting the results about our project to develop a DNA vaccine against Klebsiella pneumoniae, in which we could do a regulatory assessment thanks to the assistance of ISIDORE and the provision of the regulatory service. So now I am going to speak nothing to do with viruses, so we switch completely the topic. We speak about multi-drug resistant bacteria, which are causing 1.7 million deaths related to infections worldwide every year. And if we move to the hospital setting, the stays are extended twofold when the infections are caused by one of these pathogens. Altogether, the economic impact of this type of infections rises up to 3.5 billion per year. So, to solve this situation, the World Health Organization has encouraged to pursue different strategies, and among them there is the development of vaccines to prevent this type of infections. Compared to antibiotics, they prevent the establishment of the infection, but also reduce the mortality and morbidity associated to the infection. They provide herd immunity, and the protection lasts longer. That's why this strategy has been encouraged to be pursued. You might be wondering why we selected Klebsiella pneumoniae. The reason is because it is among the top 3 species causing antibiotic-resistant infections, both at the hospital level, but also in the community. For you to have an idea about which kind of numbers we are speaking about, in 2021, 29 European countries reported infections by Klebsiella pneumoniae, which affected over 40,000 people. If we zoom out at worldwide level, approximately 40% of the infections caused by these pathogens are by strains resistant to last resort antibiotics. So, considering altogether, Klebsiella pneumoniae resistant to antibiotics are a widespread clinical problem. And nowadays, there are no vaccines available against this pathogen. We are not the first ones trying to develop a vaccine against Klebsiella. We performed a review of the scientific literature involving this subject, and we found 75 attempts of developing a vaccine, all using different kind of vaccine technologies, from whole cell vaccines to vaccines based on recombinant proteins, but only one study was focused on using nucleic acid vaccines, despite the advantages that this type of vaccines have, like their high scalability and manufacturability, that they can be easily distributed, and also the flexibility to be adapted to target other microorganisms. We were attracted by these advantages, and that's why we decided to try to develop a DNA vaccine against Klebsiella pneumoniae, which we named Klebsigene. Here you can see the pipeline of the development of this vaccine. We are still in the preclinical phase, just in the proof of concept stage. And we are gathering together enough in vivo data to have robust data to support the patent filing of the vaccine. In our pipeline, we included a regulatory filing, but we were aware that it is important to get in touch with regulatory agencies early in the development phase, so we are aligned with the requirements since the beginning. So that's why we knocked the door of ISIDORE, because we are in academia and we don't have a regulatory department devoted for that. So we ask a provision of a regulatory service. The objective of our project was to establish the regulatory roadmap of Klebsigenes from proof of concept to product development, with 3 specific aims: to identify the regulatory applicable to the product, to perform the initial regulatory assessment, and establish the milestones in terms of regulatory that we have to accomplish during early phase of technology development. We organized the project in 4 work packages. It was granted last autumn, and since then we have been able to complete work package 1 and almost work package 2, which involve determine the regulatory field and review the protocols that we are doing, whether they are aligned or not with the regulatory requirements. And this is what I'm going to share with you today. But first, I would like to introduce to you how we were working to develop this vaccine. So here you have our pipeline. We selected the antigens of interest. We performed some in silico checking to assess that the antigens that we selected were immunogenic. There was no risk of cross-reactivity with human proteins. And so, if so, we move forward to construct the plasmidic DNA vaccines. We used E. coli to produce them. We assessed the constructs by PCR and Sanger sequencing, and then we transfected human cells to assess that the antigenic protein was properly expressed by eukaryotic cells. When we did all these validations, then we moved to produce a mid-scale quantity of the vaccine to start with the in vivo efficacy testing. So we selected 8 surface proteins of Klebsiella pneumoniae to be used as antigens. It covered 3 receptors of siderophores, 1 fimbrial subunit, and 4 porins that were related to the virulence factors of Klebsiella. They had essential cell function and a conserved sequence among different Klebsiella pneumoniae strains. We modified the sequence to be expressed in human cells and fused it with immunostimulatory elements, and all was cloned in PIVX1 plasmid. So we obtained positive clones for all the 8 vaccine candidates, and then we tried them in cell culture, and we also obtained expression of the proteins which we assessed by Western blot or real-time qPCR, and we observed different amounts of expression depending on the construct. In the in vivo phase, what we did was vaccinating groups of 10 mice with 3 doses of the vaccine. Each dose was administered 2 weeks apart. And after that, we did a challenge with Klebsiella pneumoniae and monitored survival for 2 weeks. Also, we collected blood samples to assess the antibody titers after each immunization. Here you can see the results of 2 of the vaccine candidates, the one encoding a fimbrial subunit and a porin. And we saw different performance depending on the antigen encode, with the one from the fimbrial subunit performing the best. And we see that the IgG antibodies were elicited after the 3rd immunization dose. At this point, it was when we contacted Mr. Ben Eisel from ISIDORE, who helped us to determine that the regulatory field applicable to Klebsiella was that of DNA vaccines, and he provided us with different documents related with the regulatory applicable to the set. And we went through this documentation to see which were specifically all the requirements and check which were already taken into account and which ones were missing. And here I have summarized for you the result of this analysis. We were safe in the way we were recording all the components of the vaccines, the protocols, the description of the plasmid, the genotype of the bacterial cell used to produce them, and including an endotoxin test. It was also okay that we were testing by real-time PCR the expression in cell culture, and that we were doing the in vivo analysis and testing the antigenic specific antibodies. And we could also check that the requirements for cytokines, prime booster strategies, and autoimmunity were not applicable to our product, while we were missing many things. For example, we were missing to fully annotate all the ORFs in the plasmid, to check the genetic stability of the plasmid, the percentage of supercoil plasmid, and correlate it with the efficacy of the vaccine in the in vivo test. The way we were doing the endotoxin test needs to be changed, and as well as, thank you, we need also to assess the mode of action of the immunostimulatory elements. In the in vivo testing, we need to measure the cell-mediated immune response, as well as the persistence of the plasmid and the biodistribution. This is very important for us to know at this moment because we are early in the process. So we can now address the experiments to include all this information, and this means that then later on we won't have to do them again to have this data. So at this point we are looking for partners to help with this part. So if you are interested, just let me know. And to finish, I would like to thank all the colleagues that make this study possible, the funding agencies as well, and to all of you for your attention, because you hold on until the very end, which is very much appreciated. And if you have any questions, I'll be glad to answer now or later. Thank you.
Thanks very much. Questions? It's the end of the session. People are getting tired. So, Martin.
So maybe more of a speculation question. Why do you think there are still no vaccine for Klebsiella at this moment?
At this moment, I don't know, but the previous attempts failed because there is a lot of variability in the capsule, and most of the previous attempts were focused on doing this kind of vaccines targeting this part. So this was a problem.
Thank you.
Thanks.
David, the microphone will be coming.
Great talk. I'm just delighted the regulatory advice helped out. This is a shameless pitch here. You mentioned immune response studies. You still have time?
I know,
yeah,
Patricia mentioned.
Okay, good. Just
thanks.
Okay. Thank you very much.
Thanks.
So I'm not going to delay you very much. The aim of today was to demonstrate that ISIDORE has contributed across the broad spectrum of services to the development of different medical countermeasures, diagnostics, approaches to understanding infectious disease. And so I'd like to thank all the speakers who did indeed demonstrate that. I think it's been a success as far as that's concerned. I'd also like to thank our keynote speakers, the beneficiaries of the services, not only those who gave the talks, but also the people who gave the posters. I'd like to thank the arena staff who organized the meeting, and also the arena staff who've been so actively involved in coordinating ISIDORE. For those of you who've benefited or are involved in ISIDORE, you'll know there's one person who's not here, that's Claire Connellan. It's because she's on maternity leave, and so she's been one of the people who's been instrumental in making sure that despite its complexity, despite its size, it has run in a very smooth manner. So I would also like to thank all of those participating research infrastructures and service providers who've made all of this possible. So before you rush away, we're hoping to have a photo. So I suggest that everybody comes to the front, and then after we've done that, you'll be free until the networking event, which is happening at 7:30, 2 floors down. So you can go down the stairs by the reception. So thank you, everybody, and if you can come to the front for the photo.